Oh, yeah, that's better. But you've all heard that I greeted you, ladies and gentlemen. So today we celebrate the 200th birthday of Gregor Mendel. His work laid an important foundation for the scientific understanding of inheritance. Regrettably, our colleagues in the 19th century did not elect Mendel to the Leopoldina. It's clearly one of those who is forgotten. It took about 4 decades after his death before it became clear what a great scientist Mendel was. To recognize and honor this giant of science on the occasion of his 200th birthday, a group of Leopoldina had the idea to invite all living winners of the Mendel Medal, which has been awarded by the Leopoldina since 1967. They proposed to organize this symposium that should combine the memory of a great researcher with the recognition of Leopoldina members who have made outstanding and pioneering work in the field of general biology. The Leopoldina Presidium was very enthusiastic about the idea of somewhat an ad hoc Mendel birthday symposium. And I would like to express my deep gratitude and thanks to Vice President Ulla Bonas, Regine Carmen, Christiane Nüsslein-Vollhardt, and Wittmar Tanner for their great idea and the commitment to this symposium. I'm very happy to give a warm welcome to all winners of the Mendel Medal, those who are here today and will give a talk, as well as those who will follow the symposium via a live stream from home. I would also like to extend my welcome to all participants of the symposium. I'm sure that our symposium will be an inspiring mix of a historical retrospection and cutting-edge science. Today seems to be the hottest day of the year in Halle. This may be another good reason for spending the afternoon in the relaxed atmosphere of this cool lecture hall. Without further ado, I would like to thank you for your attention, ladies and gentlemen, and it is now my pleasure to give the floor to Regine Carmen, the winner of the Mendel Medal in 2011 and one of the co-organizers of the symposium. Regine, thank you very much. The floor is yours.
Okay, also from my side, a very warm welcome. And I thought I'd rather tell you how we ended up organizing this meeting. So I was in September last year, and we celebrated the 80th birthday of Ernst Ludwig Winnacker in Munich. And we ended up sitting outside. It was very nice weather. And there was Janni Nüsslein-Vollhardt, Rolf Tauer, Wittmar Tanner, and I. And Janni asked, and Wittmar Tanner actually said that he had just finished writing an essay about Gregor Mendel on the occasion of his 200th birthday. And Janni asked, talked about what the National Academy was doing to celebrate this, and asked, so what is the Leopoldina doing? And I said, I don't know, and decided to ask Ulla Bonas, the vice president. And I called her up, and I can only tell you, you should never do that if you don't want to have additional work. So we ended up coming up with the idea of just asking all the previous medalists to come to Halle on this occasion, and to ask the people who were sitting on the table with us in Munich at the time to also come. So this is why we are all here, and I think it's a wonderful occasion to meet and to talk about science, look back, but also look ahead. So it's also for me a very great pleasure to welcome the additional guests, but I would also like to extend a warm good afternoon to all those medalists who, for whatever reason, failed and could not come to Halle, but they will be sitting hopefully in the cellar where it's bearable in terms of the heat and listen to the talks this afternoon. So I'm particularly happy that Vidmar Tanner will open our symposium to talk about inheritable elements, these elements that Mendel discovered. Vidmar Tanner is special because he has a very special connection to Mendel. He was born in Wagstadt. Wagstadt is only 25 kilometers away from Heinzendorf, where Gregor Mendel was born. So, Vidmar, I would like to ask you now to give your presentation. And after that, I think that will be a wonderful introduction to what happened, well, 180 years ago. And this will then be followed by more today's science. Thanks, Vidmar, for coming and joining us in Halle on this occasion.
Sehr geehrter Herr Präsident, liebe Organisatorinnen, geschätzte Kollegen, verehrte Gäste, the Czech people are quite humorous, so they also celebrate, actually on the very day. And they had a special 7-meter green pea. Let's see if we can do this. Seven-meter cross-section green pea was going through all of Brno, and people were interested what it is, so they finally probably got interested in Mendel and learned about him. I was thinking what I should be talking about, and I decided not to talk about the 3 rules of Mendel and about the pea experiments, because I'm assuming you all know this anyway. So I would like to talk about what you may or may not know about Mendel's life. And I would cover things like life in the time of feudalism and what actually motivated Mendel. This enormous motivation. He was having classes with more than 100 pupils every day, and as an assistant teacher. And at the same time, he was crossing thousands of peas and documenting this. Then I would like to make a point about a long discussed feature. Did he cheat or did he not cheat? And was it just lucky by choosing 7 traits? And finally, I would also like to point out that if somebody dies 16 years before he got world famous, this creates a special problems for biographers. And finally, Mendel's grapevine, he also crossed grapevine, came back to Brno via Tokyo. So these stories I would like to cover. As Regina already said, I was born in Heinzendorf, just next to next to Heinzendorf in Wackstadt. So here you can see where the whole talk. Okay. So here is Heinzendorf, and next to it is a little town, Wackstadt. This is where I came from, and actually here is Olmütz, and here's the Polish border. In 1985, I was invited by Brno University as a guest lecturer, and Nečas, the biochemist, took me around in the country, and so we passed by Mendel's house, of course, and then he was nice enough to also go to my house, and of course the main difference of the two houses was that in mine was no memory plate yet. Yeah. Okay. Johann, later Gregor Johann Mendel, was born in July 1822 to a poor farmer's family. At that time, his father had still to do unpaid compulsory work 3 times a week for the landowner Countess Waldburg. Johann was an excellent pupil. His teacher, as well as priest, convinced the parents that they should let him go to secondary school. But it was very difficult for Mendel. He went actually to Opava, or the Troppau, for 7 years, and later also to Olmütz. But it was very difficult for him because he had to study, of course, at the same time he had to tutor so that he could make his living. He got very little support from at home. With 21, he wrote in an autobiographical note, "It is not possible anymore to stand this effort any longer. I feel compelled, therefore, to enter a state that takes care for my bitter food problem. My circumstances decided my status choice." In 1843, he joined the Augustinian monastery in Brno. He studied theology and was ordinated 1847. Thereafter, he was for a short time working as a chaplain in a hospital. Since he himself suffered extremely under the sick and dying people, his abt, Cyril Knapp, decided to take him out from his priestly services. Excuse me. And he sent him as an assistant teacher to Znaimo. This is Znaim or Znojmo. This is a town between Brno and Vienna, about halfway. And here he was teaching for 1.5 years. And when he left Znaimo in when he left Znaimo, he became actually a certificate of good conduct. And I will read it to you because you will see how times were in these times. He has never used inappropriate or objectionable words. He has no other company except that with colleagues. He restricts his going out to the local teacher club together with other city notables, with the exception of 6 times visiting a theater performance. However, always together with colleagues. You see, Metternich did it as well as the Stasi. I mentioned this extreme motivation of his, and it was always asked how come that he could work so hard and be so much so extensively motivated. He was teaching from 1854 to 68 at the Brno Realgymnasium, aged again as an assistant teacher, mainly for physics and natural history. He had classes of more than 100 students. The year where he had most pupils was 117 in one class. When 2 years after he stopped teaching, the school law of Austrian Empire reduced the maximum rate to 60. In the time from 56 to 64, he carried out his experiments. He crossed more than 24,000 pea plants and managed this heroic bookkeeping of the results. How can anyone have such a power and motivation for carrying out this workload? A botanist of Vienna, Rosalie Wunderlich, published an interesting hypothesis. She speculated that one reason for Mendel's behavior might have been related to his failure in the state exam. He had studied 4 semesters at the University of Vienna, among others, with the goal of subsequently become a full teacher with a full salary. There is something mysterious about this examination. Firstly, it is not understandable that Mendel, with his intelligence, would fail at all. And secondly, no protocols and no documents of the examination exist. So Wunderlich speculates that it may have come to a controversy during the examination between Mendel and his examiner, Frenzel, who was known to have a quick temper. They may have disagreed about the generally accepted preformation theory of sexual reproduction. This assumed that the embryo was completely preformed, either in the sperm or in the egg cell. There were 2 schools fighting about this. The respective partner is thought to only independently contribute to the progeny. Indirectly contributes to the progeny. Frenzel represented this theory. Mendel did not believe in it. Wunderlich based her hypothesis on 3 bits of evidence. Firstly, immediately after the failure of the exam of Mendel, he started right away with preparing the pea experiments. back in Brno. Secondly, he carried out all crossings reciprocally. Each trait could either be contributed by the pollen or the egg-delivering plant. Thirdly, in his famous 43-page paper, Mendel only once used a footnote to stress and explain something in detail. And the English version will be here. I would like to read the German words of Mendel himself. Wäre der Einfluss des Keimsackes, gleich der Eizelle, auf die Pollenzelle nur ein äußerer, wäre derselben bloß die Rolle einer Amme zugeteilt, also der Pollen in der Umgebung, das Eis, wobei die Ei nur die Amme darstellt, dann könnte der Erfolg einer jeden künstlichen Befruchtung kein anderer sein, als dass der entsprechende Nachkomme ausschließlich der Pollenpflanze gleichkäme. Dies haben die Experimente in keiner Weise gezeigt. Whether Wunderlich's thesis that Mendel got especially motivated by an unfair and incorrect examiner is right or not, Gregor Mendel most convincingly provided the preformation theory to be incorrect. Did he cheat or did he not cheat? Whenever a few biologists get together or some physicians get together and the discussion turns to Mendel, you can bet that someone will say in this circle, isn't that the guy who cheated with his data? And this was really based on a publication of 1936 by Roland Fischer, who came to the conclusion that in Mendel's data were too good to be true. Among the very few who defended Mendel was Sidney Brno, who wrote, "I am certain he did not invent numbers, but he certainly knew when to stop counting." In the meantime, a team of English and Dutch scientists, experts in the fields of history of science and of statistics, published a paper 3 years ago in Hereditas, which came to the conclusion a comprehensive re-examination of Mendel's segregation ratios does not support previous suggestions that they differ remarkably from expectation. I do not want to pretend that I fully understood that paper. However, I read a recent review who referred to this publication, and this had ended with the line, "This is allowing us to consider this debate closed." I doubt it. Rarely are revocations very efficient. Okay, he did not cheat, but wasn't it just lucky, sheer lucky, in the sense of, hatte er nicht Dusel, so würde man in Deutsch sagen. That he only studied 7 traits, because later it was found that in the haploid genome of P, they have 7 chromosomes. Again, this is claimed that he was lucky, extremely lucky, by some of the most recent articles celebrating Mendel's birthday. Again, it is wrong. In 1975, Stixt asked, why didn't Gregor Mendel find linkage? And published the answer in Nature. He mapped all genes in the P genome Mendel had worked with. He showed Blixt showed that the 7 traits are located on 4 chromosomes. And Mendel did not find linkage because the location, those located on one and the same chromosome were so distant that they would mimic non-linkage. Thus, he was lucky with his choice, but we shouldn't call it Dusel, which always has the flavor of being undeservedly lucky. In the 20th of the last century, Thomas Hunt Morgan indeed showed that Mendel's third rule, which reads, different genes are independently transferred to progeny, is only partly correct. Morgan showed that in Drosophila, with his 4 chromosomes, the genes could be ordered into 4 linkage groups. In this moment, the chromosome theory of inheritance was firmly established. Of course, Mendel did not know anything about chromosomes. Nevertheless, he postulated that the formation of germ cells, the genetic elements, have to be segregated, and thus we predicted the reduction of division of chromosomes. As Peter van Dyck stated it in Nature Genetics of the last week issue, Mendel was decades ahead of his time. His present deduction of paired elements that break up their association and separate in different daughter cells is what makes him the founder of genetics. Yeah, I'm trying now to tell you about that this late getting known of Mendel was, of course, a big problem of his biographers, mainly of Hugo Iltis, who wrote the first extended biograph. Mendel presented his results in 2 lectures in Naturforschung der Verein Brno in March 1865. In the protocol of the session, it was written, no questions, no discussion. Unfortunately, the situation remained in principle like this for another 35 years. His publication with the modest title Versuche über Pflanzenhybride in the following year in den Verhandlungen des Vereins were rarely read and definitely never understood before they were rediscovered by Correns, de Vries, and Schermak in the year 1900. It is certain that Mendel must have been quite disappointed, especially that he could not get through to Nagaly with his arguments. Nagaly, professor in Munich, would have had the background and also the mathematical knowledge to understand him. Despite being ignored by scientists of his time, Mendel was content with his work and his life. Shortly, a few months before he died, he wrote in a letter, mir haben meine wissenschaftlichen Arbeiten viel Befriedigung gebracht und ich bin überzeugt, dass es nicht lange dauern wird, bis die ganze Welt die Ergebnisse dieser Arbeit anerkennen wird. Und es hat noch 16 Jahre gedauert. One rarely considers the consequences when a person like Gregor Mendel dies, someone whose fame is not yet recognized, least at all in the monastery. His estate, his handwritten correspondence, and large part of his library was thrown away or burned carelessly. Hugo Iltis, who wrote the first extensive biography of Mendel, described in 1924 in a book with the title Gregor Johann Mendel, Leben, Werk und Wirkung, how difficult it was to get information about the now world-famous scientist. At least he was still able to talk with contemporaries. As Jew, Iltis had to leave the country in 1939, whereby Einstein, who knew his book, helped him and his family to get a last-minute visa to the United States, where he stayed the rest of his life. So what's about that grapevine of Mendel? Mendel had many interests and intensively took care of them. He studied the weather and reported the data measured in Brno daily to the Central Registration Office of the Empire. He did all the measurements himself, including temperature and humidity. He also measured ozone content in the air up to 3 times a day, following a newly developed procedure. He was drawing maps of sunspots and changing sunspots changing with seasons, and carefully recorded the groundwater level in the year 1865 to 1880. Again, by carrying out the measurements himself. In addition, he did not neglect biology. After he had published his pea experiment, he still did crosses with many different plants, amongst others with grapevine. In 1913, Professor Miyoshi from Tokyo visited Brno. The biological societies of Japan had donated a considerable amount of money to the Mendel monument unveiled in 1910. One has to say that the Japanese were extremely interested in Mendel and were more or less the first nation who showed this. Miyoshi met Iltis, and a dedication was offered and cutting from Mendel's grapevine as thanks for Japanese support for the monument. Excuse me. not corona. The cutting was brought to Tokyo, where it was growing excellently, and it is now more than 100 years old and needs considerably propping up. The 6 pillars can be seen holding the plant up here, growing from here. In Brno, however, all Mendel's plant materials were just destroyed after the Second World War, when Czechoslovakia became a vassal state of the Russian Empire. Lysenko, strongly supported by Stalin, declared genes as non-existing and taught that only acquired characters are inherited. Mendelism became a shimp word, and his statue was removed. Ingo Schubert, who studied in the DDR, told recently that it took until 1964 that Mendel and his work were allowed to be mentioned in his gymnasium. So DDR was, of course, also under this pressure. After the Velvet Revolution, 1989, a cutting of Mendel's grapevine was sent from Tokyo to Brno, and where it started to grow a strong plant. I learned this story from Professor Nagata, director of the Botanical Garden, Tokyo. He also told me that Mendel wine was produced and from his plant and sold to enthusiasts. Thus I shortly wrote to him whether we couldn't get a few bottles of this wine so that you can drink here on our symposium with Mendel's wine to his honor. But unfortunately, Nagata wrote that they stopped producing this wine, so it wasn't possible to bring some. Since then, I also heard, however, that the wine tasted anything but good. So we may not have missed anything. Thus, let us toast with good Leopoldina wine to Gregor Mendel, to a unique scientist who admirably managed his difficult life. Thank you for your attendance.
For sharing with us insights most of us didn't know. That was really wonderful. Thank you so much. If you have direct questions, you're most welcome to ask them now. Otherwise, I refer you to the coffee break. Okay, I don't see any questions, Widmar. This will come later. Thanks again.
Yeah. Like Mendel, no question, no discussion.
So I hope you're not as disappointed as he was. Okay, thank you. And we now move on to the scientific part of our symposium. And the first speaker is Herbert Jäckle. And it's really my pleasure to introduce him. So he's from the MPI it has a new name, Multidisziplinäre Naturwissenschaften in Göttingen. Herbert received the Mendel Medal in 1999 for his discoveries concerning the induction of segmentation processes during embryogenesis in Drosophila melanogaster. Now, more than 20 years later, he will talk about lack of histone synthesis causes cell cycle arrest during early embryonic development. So he stayed with Drosophila, but I think the depth of what he's doing now is very different from what he is doing was doing 20 years ago. No? Okay, then convince us. Thanks for coming.
Okay, dear colleagues and friends, dear President, usually I forget birthdays. And if I'm not reminded by my wife, I would even forget my own one. However, I think in the future, I will not forget Mendel's birthday anymore, because this morning at 4 o'clock, our second grandson appeared on this planet. So he is now 10 hours old, so I hope in the future he will not develop into an absent sailor, but if he would become a scientist, I wouldn't be unhappy.
Congratulations.
Thank you. Well, it's not. There are problems. This is when you go from a Mac to a real computer. I'm sorry. So anyhow, I can start. I mean, for me, personally, Mendel was always a mystery because I never had a genetic course. However, there was a paper in 1980 co-authored by Janine Volhard and Eric Wischhaus, and that basically determined my whole scientific life. And I have a little picture. It doesn't work. I see it here. Oh, now it's there. It's okay. It looks okay. Yeah. So this was, for me, this life-determining discovery that genes determine the body pattern. And, of course, of Drosophila melanogaster, which was sheer luck, or it wasn't sheer luck. Anyhow, what the basic message was is genetics and phenotypic analysis show that the body pattern of Drosophila is generated by maternal and zygotic factors, and they subdivide the anterior-posterior and dorsoventral axis by determining increasing smaller units. Now, the message is very simple. At the beginning, there are gradients, and they determine basically the expression domains of the genes at the next level, the gap genes, and they act as real neighbors. They try to suppress each other, so they have distinct domains, and at the end of the day, all these work together in a hierarchy to determine a metameric pattern of the so-called parallel genes. Now, this was the message of that paper, and what we did then, or what we started, is that we looked into these genes, the molecular basis of it, and found out that they are basically transcription factors. And depending where you are in a certain position, you have a combination, or better here, you have a combination of these genes, and finally, you have the metameric pattern. Now, what is also known, of course, was that if you don't have bicoid, the gradient, the red gradient from anterior to posterior, you have no head and no thorax. And this is basically the end of my introduction, as well as the end of the textbook story. Now, our question first was, what happens if bicoid is expressed throughout the embryo? And you see it here. There is, of course, more in the anterior region, because this is a wild-type embryo, which has a transgene expressing picoid throughout the embryo in a basically at a flat level. And the prediction is that because picoid is suppressing, quote, its neighbor, as I said before, is that caudal in the posterior, the green curve, the translation fails. There is no anterior-posterior information, and the entire embryo takes on, to a certain degree, head identity. So this is the expression again, and you can see the prediction is true. Caudal is completely gone because it is suppressed by bicoid. And the zygotic segmentation genes, at least at the level of the gap genes, are extended into the posterior. And the idea at that point for us was that there could be additional regulators which fine-tune and set the spatial limits of the bicoid-dependent genes. And of course, the question is always, who is it? And to make a long story short, there are genes, or torso is a key component of the terminal regions, anterior and posterior. And the torso is controlling another gene and the expression of the gene, which is called Capicua. Not only Capicua, but I would like to show the system here with this one gene. And torso signaling simply expresses Capicua activity at the poles, as depicted here. So the question is then, how does that work? And the answer is simple. Capicua encodes a DNA binding domain, so it's a transcriptional repressor, and it binds to a certain region of the promoter regions, which is defined, of course, by the sequence. Now, we took a head gene, which is under the control of bicoid, which is expressed in the anteriormost region, makes a stripe here, and this is how it is normally expressed, and we simply took a transgene which contains this enhancer and drives LAC-C as a reporter gene, and you can see basically the same pattern. So we looked into this in more detail and asked the question, can bicoid and Capicua interact on this enhancer? And if so, does it affect the expression pattern? And the answer is relatively simple. Yes, they can interact. So if you take 3 bicoid binding sites, and this is artificial, and express it, and express a reporter gene, then it goes up from the anterior to a certain position in the embryo. If you now add, for example, 2 Capicua binding sites to these bicoid binding sites, in that case, you get a shift towards the anterior. Now, if you take Capicua away, it moves back again. And this clearly indicates there is a binding of Capicua, and the promoter is co-regulated, so to speak, by Capicua. So we have a simplified model. We have, of course, bicoid, which is activating, and you see here the enhancer region, and it's on when Capicua is low and repressed by torso. Now, if you take torso away, in that case, Capicua is active, and torso is not active in the central region of the embryo, and thereby Capicua binds and prevents the activity of bicoid to turn on the gene. And so in this case, for example, if you are in this position, you have no Capicua, and so on. So the torso-dependent Capicua determines the precise spatial limits of the activating bicoid activity. So in total, there are 5 binding sites, which are decisive as a promoter region. And this is all within 71 base pairs. And a transgene containing these act in all chromosomal positions that we have tested. So that means the nucleosome positioning, histone modifications, and regulatory chromatin components must be properly in place. Now, coming to the nucleosomes, I mean, you all know it's basically a core around DNA is wrapped, and it's composed of the histones H2A and H2B, a dimer, and histone H3 and H4. In addition, these histones contain so-called N-terminal tails, and they can be modified or replaced by phosphorylation, ubiquitination, acetylation, methylation. So there are basically modifications that are important because depending on who is modified and which way they are modified, this attracts additional factors to the chromatin to cause silencing or opens the DNA for business and changes, for example, the distribution of the DNA within the nucleus and many more functions. So coming to the histones, to the evolution, bacteria don't have histones. If you go to archaea, single histone-like genes. If you go to yeast, there are few histone genes, and when you go to real animals, then there are many histone genes, and humans are filled up. The only problem is, how can you do genetics if you have many histone genes? And there is, again, a superb situation in Drosophila, meaning that, for example, Caenorhabditis elegans, the worm, has 66 histone genes, 11 loci. It's very difficult to knock out 11 loci. If you go to flies, there are 46, and there is one locus. And if you're lucky, and it took us 4 years, then you can basically generate a deletion of all histone genes at once. And the result is, of course, that you have, quote, a histone-less mutation, which, of course, would somehow not survive. But as a heterozygous, it survives. So you can basically do the Mendel one. 1/4 of the embryos of heterozygous parents who survive, are homozygous, and they lack the histones, as you can see here. This is wild type, a staining. This is the mutant, staining 2, but nothing stains. And you also can look in this situation, and you see histone 3 is missing in the position where you normally expect it. So there is no histones, and these embryos, unfortunately, or naturally, die before they form a cuticle. So if you don't have histones, there is no cuticle. If you take 2 copies of histones with a transgene, you get a certain, you get a kind of cuticle. Of course, these embryos are dead as well. No real head. If you add 6 copies, the head is coming up. And if you have this 46, then they are there completely normal. Interestingly, the developmental genes, which I talked at the very beginning, are, quote, normally expressed, but at a lower level. That's all. But the histone mutants arrest in cell cycle 15. And this means this is the cell cycle. You have mitosis, you have an S phase, you have a G2 phase, and then it stops. And you can see that with cyclin B, which needs to be degraded in order to jump from G phase 2 into mitosis. And you can see in these embryos no degradation of cyclin B, so mitosis is not initiated. So what is special about this cell cycle 15? If you look what happens in the beginning, there are 13 nucleocycles, and they are all guided by maternal factors. So we have maternal histone supply, messenger RNA, and protein. At this cycle 14, we have first-time zygotic transcription of the histone genes, and we have, in addition, a G2 phase before going into mitosis. This takes 20 minutes, rapid nucleocycles without G2 phase. This is 90 minutes or something like that. So, I sum up to here, is there is absence of zygotic histone supply causes cell cycle arrest sharply after the G2 phase in cell cycle 15, and it causes embryonic lethality. What I have not shown, but I assure you, DNA replication is slow, but absolutely complete. DNA repair is not activated, so there is no checkpoint at that level, and there is also no apoptosis and no necrosis in such embryos. And the conclusion is very simple. Zygotic histone supply is essential. And the question is, does this, of course, affect nucleosomes? And if so, how are they affected? Now, a short recapitulation. So we have the replication of the DNA. The histone go off of their loci, and they reassemble in combination, of course, normally with newly synthesized factors. So what happens when the zygotic histone synthesis is lacking? You can see it here. So the distance between the nucleosomes is enlarged, and the nucleosome occupancy is decreased. So this interdiet, and here is the result in his mutants and in wild type, and you see it's enlarged or decreased. So the question is, or was then, is the chromatin accessibility pattern, has it changed, and how does it look, for example, in the his mutants? Because remember, these developmental genes are properly expressed at that time. And what you see here is the wild type situation and the accessibility, and you see the peaks always in the 5' region of the genes, and this is basically maintained but strongly reduced by a factor 4 to 5. And this is now one chromosomal region, but we looked in many chromosomal regions, and it's all the time, quote, the same. Reduction, but in principle, the pattern is okay. Now, how about the nucleosome positioning at or downstream of the transcription start site in the his mutants? And it's altered, as you can see here in blue, the wild type situation, and in green, the mutant situation. And you see the height is very different, and it doesn't sharply turn off as we see it before. Now, what is also important is, of course, how does it affect gene expression patterns? And what you see here is that a total of 3,000, close to 3,000 genes get up, are stronger expressed, and about 1/10 of this value is decreased. So obviously, it's easier to transcribe genes. And what is so special about these genes? When you look at them, who is who, what we found is that many of these genes are not expressed in wild type in early stages, but at later stages, and they come up in very peculiar kind of time points for example, meiotic genes come up, never seen in an embryo. Spermatogenesis genes come up, never seen in a wild-type embryo. Protein synthesis pathway is completely conserved. And they come up, and it does not make, quote, any sense at that time. Now, what happens? If you look in wild type, the RNA polymerase 2 is, of course, on the transcription start site, and it is gone at the transcription end site. In the His mutants, it's different. It's mainly in the gene body rather than at the transcription start site. So this basically says that the transcription is initiated not only at the transcription, at the, quote, normal transcription start sites, as you can see here. Normally, you would find 100% in the promoter region. In these mutants, more than half are initiated in different positions, such as introns, intergenic regions, exons, and the untranslated regions, which you find in the messenger RNA. How about the epigenetic marks? As I told you before, they are absolutely essential for guiding, basically, transcription. And interestingly, the activating histone marks maintain in their position, but again, they are strongly reduced. And this basically explains why, at the time, these developmental genes are properly expressed. Now, what happens to the repressing marks? And they maintain their position, and they are, let's say it, cut a long story short, they are always at least as high with their profile as they are in wild type. And this, again, is not only true for this region, which is demarked here, but it's true for all the regions we have analyzed, and that was quite a lot. So the summary: modified histone nucleosomes have a positional memory. There are active marks which are maintained but reduced, and repressing marks are maintained and not necessarily reduced. So the chromatin landscape is maintained, but there is increased chromatin accessibility in addition, meaning we do have transcription start sites within the gene bodies. Now, why do cells, and that was the key question to us, of course, why do cell cycles stop sharply during cycle 15? And for that, we have done many stainings with key cell cycle markers. One is string, which is the CDC25 phosphatase, and cyclin B. And cyclin B, just to remind you, is built up in the G2 phase, and when string comes up, it is degraded. So there is an interplay between the two proteins. So we have an inactive cyclin B complex with a phosphorylation site here, and this is stolen by string. And once this phosphorylation site is gone, due to this phosphatase activity, in this case, the complex is degraded and the mitosis is initiated. So string is actually an initiator of mitosis, and the result, or it acts through dephosphorylation of cyclin B. Now, how does it look? In the histone mutant, do they transcribe string messenger RNA? Is there transcription? And the answer is yes, it is. I don't show you the data, but immediately this messenger RNA, which is in the nucleus, is immediately degraded. So it is transcribed, yes, but it is not maintained as messenger RNA. And you can see it here. There is no string RNA anymore. and in wild type there is string RNA present. Now, we then figured, okay, it's transcribed, but the messenger RNA is not maintained, therefore there must be a mechanism to degrade. And the question is, how can that happen? And from all what we know is, usually there are sequences within the messenger RNA recognized by the protein that degrades them, the enzyme. And so we looked into and asked simply, is there something in the 5' UTR or in the 3' UTR or somewhere else? Now, the transgenes we have produced cuts off the intron, so it is pure cDNA or only the coding sequence of string, and we exchanged the 5' or the 3' and used again the transgenes and looked what happened. And what we used as an internal control, because we have these stripe genes, the parallel genes, we used the pair marker to express now string in stripes along the anterior-posterior axis. So we have string expression, no string expression, no string expression. And the result is cyclin B goes away wherever string is expressed. You can see it here, and it's gone here. So in this case, there we would have, quote, the wild-type situation, at least in this band. And you see what happens here. The string is expressed here, cyclin B is turned down, and that's it. But this is only true if the 3' UTR is replaced. Therefore, there must be something in the 3' UTR, which is basically telling a protein you can degrade here, and there is an interaction. And we found also that this is due or dependent on a factor which is called held out wings, and this is a dual regulator of string messenger RNA, and it encodes distinct splicing variants. On one hand, there is this how large, which destabilizes string messenger RNA, and there is a short version which just does the opposite, namely sitting there and making sure that string is not degraded. And we found two binding sites, one in the 3' UTR and one in the intron. But the intron was gone anyway, because we didn't use it in the transgene. And if we replace basically this region with another 3' region, in that case, there is no degradation. And the interesting thing is that this degradation due to how L is somehow peculiar, because in wild type, it is not expressed at all in wild type. But in his mutant, it is amplified. Whereas the small version, the stabilizing version, this one here, is not really affected in comparison to wild type. So from this we know now, summary, that string messenger RNA is transcribed but gets immediately degraded. Destabilizing how L is expressed in his mutant embryos, but not in wild type embryos. And the deletion of how binding site causes string messenger RNA stability and leads to a rescue of mitosis. And here we are now, again, in a situation to ask the question, why? And so for the rest of my scientific life, I would like to see who controls how, because it's kind of mystery. It doesn't come up in wild type, but it comes up in his mutants, and it must be at the level of splicing differential splicing. And we'll see how far we can get within the next few years with a small lab. But I wouldn't stand here without all these people who contributed to one or another aspect of the work. In particular, those two people who did all the work with a histone. Ufuk has his own group. We collaborate, and we have a common student whom we supervise, both of us. It's terrible for him, but it's okay. And for this, I would like to thank you.
Thank you, Robert, for sharing with us these really exciting results, which obviously are only possible, would not have been possible by doing classical genetics, right? Because you needed the mutant, where all genes are gone.
Right.
Okay, questions?
It didn't take 16 years, it took only 4 years. And a student very close to a nervous breakdown.
Yes.
My question goes to the rescued embryos. How do they are still lacking the histones then? So how do they then develop?
We don't know whether they develop. We only looked whether they go in mitosis, and they go at least for one additional round in mitosis.
Wouldn't it be very interesting to see how they develop without histones eventually? Because you showed us the repressive and the activated marks. These are histone marks, right? So these are probably maternally inherited histones.
Yes, these are the paternal histone marks, because you don't have zygotic ones, and these know basically where to go, which is an interesting phenomenon in its own right.
Absolutely, but I think if you could then look...
Oh, they would die. They don't have a chance to survive, I'm absolutely sure.
Yeah.
Yeah. This is why we went only for one cycle.
Thank you.
Are there more questions? Then maybe I can ask one. So I was intrigued by this experiment where you saw this transgene copy number dependent rescue. Mm-hmm. So what is actually happening there? How can this be
well,
copy number dependent?
Yeah, the interesting thing is you have at least 23 per chromosome of these histone clusters.
Okay.
So you have all the histones, and followed by all the histones, followed by all the histones, 23 times.
Yeah.
If sequencing is correct, when you have repetitive sequences. Now, if you have no copy, it's clear. If you have 2 copies, then the S phase is rather slow. And you need at least 12 copies, that's what we estimated, in order to have the normal speed and to end up with a perfectly happy embryo.
Yes.
And fly, which is fertile.
Okay, but I mean, so I'm interested in the stages in between, in terms of what kind of histones do you actually assemble in these?
This we don't know. We have not looked into 6 and so on. The idea is basically that you can progress a little bit because they don't survive as flies.
That's clear.
Only when you have 12 copies, they survive. And why they die at different stages, we don't know. We haven't asked the question.
Okay.
My student would say, it's so much work.
Yes, I would probably not knowing it, but I would intuitively agree.
Yeah.
More questions? If not, I refer additional questions to the coffee break. Thanks again. It's now my pleasure to introduce the second speaker, Nick Barton, and he's from the Institute of Science and Technology, Austria. It's an institute located in Klosterneuburg in Austria. He received the Mendel Medal in 2013 for his work on narrow hybrid zones that separate different species, and in addition for his contributions to our knowledge of how species adapt and how they split into new species. Today he will talk about using hybrid zones to analyze divergence and adaptation in nature. Welcome, and we are all looking forward to your presentation.
Thank you for the nice introduction. It's a great pleasure to be here again. So I shall give quite a Mendelian talk, I think. I'll start actually back a long time ago with my topic of my PhD thesis, which was on a narrow hybrid zone between two populations of grasshoppers. I chose these cunningly as a pedestrian grasshopper, so I could catch them rather easily. They're wingless. And I've carried on studying hybrid zones of this sort really through the whole of my career. And really, I'm just actually back on the way back from fieldwork on the system that I'll describe to you today. So this first system that I started working on, this grasshopper system, was based on a chromosomal difference, a fusion between the sex chromosome here, single X in the male, 2 in the female. This is male meiosis. And you can see the X has been fused onto a pair of autosomes. So it actually behaves as a sex-linked Mendelian trait. And what's remarkable about it is that to the south of the French-Italian border, the ridge of the Alpes-Maritimes, you find the fusion. To the north, you find the ancestral form. And this boundary, this very sharp boundary, runs for 100 kilometers east-west, but it's only about a kilometer wide. And so here you see one region where you go from 100% fusion to 100% unfused, and a smooth transition in between. And it's this kind of very sharp transition between two genetically distinct populations that is intriguing. So I've studied a lot of other systems which show the same sort of features. A classic example is warning pattern in the wing patterns of Heliconius butterflies, where in any one area in South America, different species of Heliconius butterflies, and in fact other species, will share the same warning pattern. So the whole of South America is divided into a kind of mosaic. In any one area you get particular patterns, but the changeover from one pattern to another occurs very, very sharply, again over just a few kilometers. And I've also studied a European example, the Fire-Bellied and Yellow-Bellied toads, Bombina bombina, Bombina variegata. These are really very distinct, unlike these two examples where the differences are genetically quite simple. The Bombina species are adapted to very different habitats, to breeding in permanent ponds or in temporary puddles. Yellow variegata breeds in puddles, red-bellied Bombina in ponds. Many, many differences here, but again, where they meet around the Danube Basin, around the Carpathian Mountains, there's a narrow transition from one type to the other, over about 6 kilometers. So these are very striking patterns which we'd like to explain. But what I'll do is show that we can actually find out a lot about the evolutionary processes acting in these populations and make inferences in a variety of ways about things like the rate of gene flow, the strength of selection, which would be very difficult to access directly. And I'll talk about the system that I'm working on at the moment, Antirrhinum majus, which has different flower colors, actually, in a sense, rather similar to the Heliconius system, with simple Mendelian inheritance, as we'll see. And I'll summarize a variety of approaches which together tell us about the evolutionary processes maintaining this sharp distinction between hybridizing populations. But at the end, what I'll do is actually undermine my whole talk by saying that perhaps these simple systems where we have a simple Mendelian basis for the trait variation, these are perhaps not typical, and it's actually unclear what fraction of adaptive evolution we can capture using the sort of methodology that I'll talk about. So I should, first of all, since I'll no doubt run out of time, and I should thank people at the beginning. I think this project started in the Antirrhinum system with Rico Cohen, a developmental geneticist in John Ennis, and much of the work particularly on building a pedigree in a wild population, was set up by David Field and Melinda Pickup, now in Perth, and Tom Ellis and Parvati Saranjanad are students, Tom now postdoc in the GMI in Vienna, who did a lot of the work. So the system is generally a very nice one. There are about 20 more or less named species, subspecies in the Western Mediterranean. These are sort of a variety of flower colours. Actually, they're all interfertile, and so one can do genetics, and they're actually a really classical genetic model system. And the system that we're studying is actually very limited in a sense. We're studying a hybridosome between two subspecies, Pseudomajus, which has magenta flowers, Striatum, which has yellow flowers, and these make contact. Press the wrong button here. These make contact about 100 kilometers northwest of Barcelona. In fact, you can sit on Google Street View, and if you look up the village of Planoles, you can look on Street View, and you can see as you go along the magenta flowers by the side of the road changing through to yellow over about a kilometer through a very narrow transition in which you have all of these different intermediate genotypes. It's slightly embarrassing for someone who likes doing wild field work that these actually really like growing along the side of the road. So the field work consists of walking up and down the road, which is maybe a little disappointing in a way. Okay, so these, as I say, were a model system from the early days of, since the rediscovery of Mendel's work. And the genes responsible for flower colour differences were largely determined 100 years ago. Rosea and Eluta are two tightly linked genes, in fact, one of the first examples of linkage to be determined 100 years back. They control the distribution across the flower of the anthocyanin pigments, the red pigments. And then Flavia and Sulfurea, mostly Sulfurea, in fact, control the presence or absence, or rather the extended distribution of the orone pigment, the yellow pigment. So roughly speaking, you can determine 6 phenotypes just by looking at the flowers, and you can do a bit better than that, in fact. And these genes have been mapped onto different linkage groups, different chromosomes, 8 chromosomes in antirrhinum. Okay, so the first approach, one which really dominates the field now since the astonishing technology that enables us to get cheap and abundant DNA sequences, this approach is called scanning the genome. You basically look along the genome for statistical signals of something peculiar happening. And this work was initiated by Rico Cohen based on pooled sequences. You take 50 plants from one side of this transition, 50 from the other side, 2 kilometers apart, and sequence them as a pool, so you can simply compare the frequencies of different single nucleotide polymorphisms, or SNPs, SNPs, as you move along the genome. So if you look across the whole genome, the 8 linkage groups here, this top row, you measure something called FST, which is a measure of relative divergence, the pairwise difference between the two populations relative to the diversity within. And you see very clear peaks. The peak on one end of linkage group 6, it's not actually coloured in red, but it almost is. It sort of stands out as the strongest signal, and something's going on there. In fact, if you look in more detail over a couple of hundred kb, you see 2 peaks, more or less, one here, something funny in the middle. We don't quite know what's going on. But these 2 sharp peaks, in fact, correspond to the classical Rosea and Eluta loci. So, we've found after 100 years, using very fancy technology, the original Mendelian genes. And in fact, Rico's group did an enormous set of crosses to really map these down. Okay, so this is fine. But actually, the interpretation of these kind of patterns is non-trivial. Because initially, when people saw these sort of patterns, they said, these are the genes responsible for speciation. They are maintained different by selection of some sort. They present a barrier to gene extent. They allow divergence. But in fact, this statistic is not a measure of just divergence. It's a measure of divergence relative to diversity. And it turns out that in this case, and in most other cases like this, these sharp peaks are driven actually by reductions in diversity, which are probably due to what we call a selective sweep, the fixation of a mutant or multiple mutants that sweep through the population. And as they fix in the population, they carry with them a chunk of chromosome, which is genetically homogeneous, which is the lucky background on which the original mutation occurred. And so we get these regions of low diversity reflecting past selection, which then, once they have diverged, and once they're maintained as a difference between the two populations, creates then a barrier to gene exchange. And we do in fact see a broader increase here, and that's statistically significant, although it takes a bit of work to show that. There's a broader increase here, which is consistent with the effect on having these two tightly linked selected differences impeding the exchange of genes in the surrounding region. Okay? So this is the kind of approach that we can take if we just use sequence data, and this is dominating the field because it's so exciting to have floods of sequence data. But we've got a bit carried away and carried on sampling over 11, 12 years now, and the main aim of the project, which I'll come to in a minute, was actually to build a pedigree to sequence enough polymorphic loci that we could determine who's the parent and who's the offspring, to determine relationships, and we're still in the middle of analysing this. And to do that, we sampled basically all the individuals we could find over 11 years, 22,000 individuals. It's about twice that actually now. And what I'll focus on now is just 9 SNPs from 5 different linkage groups, which are those that we determined were substantially different between the two populations. And I should say that if we go back to these patterns, these spikes across the genome, there are something like 7 or 8 significant spikes in this statistic. We can also look at pools of populations at 8 different locations, so we take more populations, then we ask, do we see sharp transitions over a kilometre or a few kilometres, and that is an independent way of identifying signals, and that also identifies the same loci, and remarkably actually, especially to someone like me who's rather sceptical about this stuff, actually it kind of works in the sense that these loci were all known to have something to do with flower colour a priori, so they were already had been studied as, if you like, flower colour genes, so this all makes sense. We then looked in detail at the transition over a 25-kilometre transect along this valley from one type to the other, and the rosea, luta, which are tightly linked, change very sharply over less than a kilometre. Sulfurea, which controls the yellow pigment, changes a bit less sharply. Other genes change a little bit more noisily. It kind of makes sense because those have less of a dramatic effect on phenotype. So we can measure the shape of this transition. We can see this in a kind of summary by looking at what you can call as a hybrid index. We simply take 5 unlinked regions which have diverged, and we can then make an index which is simply the sum of the scores. If they go 0, 1, 2, homozygote, heterozygote, homozygote, we add it up over the 5, we go from 0 to 10, and we plot this going from 0 to 10 on a kind of logistic scale, expanding the sort of tails, if you like. And it's not very well drawn here, but basically we've expanded the region from 9 to 10. This would be 9, 9.9, 9.99, and so on. And if you had a simple sigmoid transition, which is what simple theory predicts, then you would see a straight line. But we don't see that. We see the sharp transition over around a kilometre. If we just, pull together all of the diagnostic regions, you see the sharp transition. But you see a sort of gradual tail on either side where you get some magenta plants. In fact, you get some pure magenta plants right the way out here, 10 kilometres away. Right the way out here, you get some pure yellows. And that reflects long-range dispersal to some extent. But there is another factor in here, and that is reflected in a second feature we can get out of the data. As well as looking at the mean of this hybrid index, we can look at the variability in that index. And we see that the variability in this measure of how magenta-like, how yellow-like you are, that variability is inflated in the middle, reflecting correlations between the state of one gene and the state of another. In other words, if you are magenta-like at one locus, you're more likely to be magenta-like at another locus. And that correlation inflates the variability. We can estimate, turn it round, and from the variance in hybrid index, estimate this correlation from 0 to 1 or minus 1 to 1. And it's kind of noisy, but there is a substantial positive value in here, about 0.05, 0.04. And this simply reflects mixing. It reflects the fact that you've got combinations of genes characteristic of this population, characteristic of this population, coming together in the middle of the region. This mixing generates positive associations. So we can actually make quite a lot of inferences from the detailed pattern of genotype frequencies in these hybrid zones. We have a series of declines that coincide, that they're quite narrow, and we have these correlations. We can actually work back and infer that on a very simple kind of back-of-envelope calculation, you need a dispersal range of about 50 meters per generation. The typical dispersal has to be around that, which is in the right ballpark. Although, as we'll see from the pedigree data, we actually there have indications, firstly, of a very skewed distribution. Mostly they go less than 50 meters, but a few go a very long way. And that kind of complicates the analysis. If we know the dispersal, then from this kind of estimate, or from direct measures in the pedigree, we can actually then estimate the selection needed to keep them distinct. And the selection coefficients are quite sensitive to the dispersal range. If you've got a short dispersal range, you don't need much selection, 3%, let's say. If there's a bit more dispersal, then you need stronger selection. And so we're in the middle of really trying to get the best estimate of selection. But the point is that in these kind of analyses, and this kind of methodology that I've used on the other systems I talked about, you can estimate things that are really hard, in fact impossible, to measure directly. You can't ever measure a 3% selection coefficient in a natural population. Even in the lab, it's pretty challenging. So, the open question we have is, can we explain both the width of these transitions? Well, yes, but can we explain the detailed shape and the pattern of genotype frequencies? And that's more challenging. So I'll finish up the antirrhinum story with a direct approach, say, well, instead of messing around with all these fancy kind of statistical methods, why don't we kind of just look directly at how far the genes move and, how the numbers of offspring, the fitnesses of the different types vary. And we can do that if we know the pedigree, if we know the parents and the offspring. And this was the aim of collecting these enormous samples, started by David Field. We had 100 SNPs on these 22,000 plants. We can infer from that the relationships. And so, for example, if we take individuals in 2010, these are individuals where both the mother and the father have been inferred as parents of some individual in a later generation. And what actually really surprised us here is that the offspring are identified many years later. So actually in this distribution, you find hardly any that are offspring the next year. They're mostly 3-4 years down. And we were kind of puzzled by this, but this is simply because there's what's called a seed bank. So that the seeds, surprisingly, don't germinate straight away as they do in the greenhouse. They actually spend 2, 3, 4, 5 years, we don't actually know how long, and you see this distribution of germination times, identified from these pedigrees. So then, the actually main aim of this, and again, it's something we haven't finished the analysis, so this is somewhat preliminary, but you can just say, well, what's the chance of being assigned as a parent if you have one of these 6 different phenotypes actually identified just from the Mendelian phenotypes? We have the SNP data, but you get kind of the same picture. So if you look in the central region here, you find that the hybrid phenotypes actually are less likely to be assigned as a parent. So there seems to be, maybe, we don't know, maybe the bees just don't like the look of orange or white flowers. They like the yellow or they like the magenta. On the yellow side, you find yellow does best. On the magenta side, you find magenta does best. Again, the hypothesis, and we don't have good direct evidence of this, it's very hard to get, is that the bees prefer what they know. They train on the common phenotype, and this is known in other systems. So, summary of all this is that we can take a sort of sequence-based genome scan approach that locates the genes responsible for flower color differences. It doesn't tell us anything about the processes that make them different, but by looking at the distribution of genotype frequencies, the shape of the declines and the associations between different loci, we can get indirect estimates of selection and dispersal. And we can now, with the pedigree, get direct measures. So we have a number of different methods which reinforce each other. So, of course, it still leaves a lot of questions open. One is, how does selection actually act? The mechanism is really hard to get at. We've done lots of looking at the plants and looking at, which ones the bees actually visit. This is pretty hard to do because this is, fairly high in the mountains. There are just not many visits. It's very boring sitting watching a plant, and it's actually just, it's proved beyond us to persuade enough students to watch flowers for long enough to get good estimates, especially when the differences are not necessarily very big. Are the two populations really, subspecies or species. They look very different. But actually, if you look at the sequence divergence, it's limited to a very small region around these half dozen, 5, 10 loci. So there's very little effect. And in fact, probably these different taxa of Antirrhinum are all one biological species sharing genes. And the open question is, how far can we explain in detail these patterns through selection acting on around 5 loci? So I'll finish up by kind of undermining this story by saying that these kind of systems, which are the bread and butter of population genetics, especially these days, you can study them, you can find out a lot, it's very satisfying, but is it typical? And we actually know that it isn't, because variation in most traits, particularly most important traits such as disease risk, such as crop yields and so on, these are the stuff of quantitative genetics, and we know that they depend on small effects spread over very many different genes. And we've known that actually for a century or so, shortly after the rediscovery of Mendel's work, simply because artificial selection works, as Darwin documented in such detail. And that implies that there are not typically a few major mutations which you can pick out and fix. You select on a population, you can keep selecting, keep changing by many standard deviations, producing entirely new phenotypes, as we see in the success of domesticated plants and animals. Those have to be due to a substantial, a very large number of genes of small effect. And this has become much clearer now with genome-wide association studies simply giving up on sort of carefully controlled crosses and so on, and simply taking the whole population. We're now up to samples of 1 million or more in the human population, and simply looking for an association between a polymorphic site and a trait. And you can pick these up with such enormous statistical samples, but you find that the effects are very small. The archetype is human height, highly heritable, and yet the largest variant that you can pick out in the human population has an effect of 1% of the variance, and you really struggle to explain 20, 30% of the variance based on, many thousands of sites. Essentially, the variation in height is spread over the whole genome. So, in fact, this trait variation is consistent with what we call the infinitesimal model. This is the backbone of quantitative genetics. And this infinitesimal model essentially says that if you take a cross, the offspring of that cross will vary in a Gaussian distribution with a variance that doesn't depend on the value of the parents. And that's because there's such an enormous range of possibilities locked up in the genetic differences between the parents that the variation you see in a trait in the offspring is very small compared with the range that you can get to potentially by selecting. And this infinitesimal model works. GWAS confirms it in a sense. What does that imply? Well, an interesting connection, now this I'm more or less done here, an interesting connection comes from a result which student Michel Hledick just derived, worked with Gaspar Tacik at IST, showing that the rate at which selection accumulates information, in bits essentially, by taking the random space of possible DNA sequences and narrowing it down to those sequences that actually work, that are functional, that accumulation of genetic information one can quantify, and it's limited by essentially, it's limited by the product of population size, effective population size, and the heritable variance in fitness, V. So the increase in this information measure per generation is at most population size times fitness variance. That's actually a very large number, so selection, of course, doesn't approach that limit. But an intriguing corollary of this work is that adaptation is most efficient when it's polygenic. If you look at these simple models, you find that in order to approach this limit, you have to have very large numbers of variants of very small effect, which actually fits with our experience in artificial selection. So then I'll end with this paradox that adaptation is most efficient when it's highly polygenic, but then we're operating in this infinitesimal regime when, almost by definition, there's very little trace in the genome. So the bulk of adaptation, in fact, the bulk of the adaptation that we care about, things like disease risk or crop yield or whatever, is extremely hard to pin down by the kind of Mendelian approaches that I've described in the bulk of the talk. So I find that kind of inspiring because there's lots of scope for maths. You could find it kind of depressing. It depends on your attitude, I think. Thank you.
Thanks a lot, Nick, for this inspiring presentation. Questions? Yes.
Yeah, thank you for the presentation. I'm just wondering whether you have looked into the ecology in those hybrid regions. I could imagine that there may be an issue looking at flower color, for instance, with pollinators that might prefer one or the other
Sure.
species.
I mean, in other systems, in Mimulus, which is maybe the best studied pollination system, there seem to be different pollinators in the two sides, and certainly it's known that hummingbirds prefer different kinds of colours to bees. Here we see the same species of bumblebee pollinating all the time. There seems to be no difference in which species are pollinating. We've also kind of looked at the habitat. It's kind of hard to do because they live in disturbed habitats, so it's hard to describe. We don't think there's any systematic ecological difference between the yellow population and the magenta population, simply because if you go to different places, in one place, as in here, let's say the yellows are higher up than the magentas. In other places, it's the other way around. So the most obvious ecological variable in this mountainous region just varies. And so it's very hard to see anything systematic going on.
So you mentioned that there is a selection acting against hybrids. So this is based on incompatibility, or what is the basis?
Well, I mean, as I say, all this is indirect, and I quite like this because you can, I like doing the maths and so on, but of course it's frustrating because you don't know why it is that the hybrids have produced secure offspring.
You could do an experiment, right?
We have done experiments, but it's kind of hard. So yes, we've had some hints of this. I mean, the sort of experiments you do is to set out an array in the greenhouse or on benches outside and just look at pollinator visits. And we did this kind of in Austria with bees what we bought from Holland. So it's not, not quite nature. And then, of course, we do have plans to try and do it out in the field, but then you don't want to disturb the natural situation. So we think it is about the bees not liking these flowers, but we don't actually have direct evidence of that.
Okay, I don't see any pressing questions right now. Then I can release you to the coffee break. The break, you can get coffee and water and soft drinks downstairs. And you also have the opportunity to actually see the exhibit by Herlinde Köbel, Fascination About Science. And there will actually be a guide taking you through this exhibit if you are interested. So I think we meet again maybe in half an hour, is that? Maybe 25 minutes. Yeah. Yeah, 3:10. Thank you.
So welcome again to the second part, or session number 2. I'm still a bit out of breath because shuttling 2 times between here and the parking lot at 80 degrees centigrade is at least for me a major challenge, or has become a major challenge. And Gregor Mendel, by the way, would probably be pleased. Vidmartana mentioned that he has been taking weather records and squeezed out 9 publications out of those to see this extraordinary meteorological condition today here in Halle. Right, but let's get back to science. Dear President Haug, dear Vice President Ulla Bonas, Honorable Mendel medalists, dear guests here in the lecture hall and online, as far as I understand. We have guests also following the session online. It's my great pleasure to take you through the session. I am Andreas Grana and I'm a plant geneticist. And like many colleagues of mine, I have been mesmerized by the work of Gregor Mendel, and in particular by its historical context that has been discussed already in the lecture of Vidmartana, and therefore I can skip some of the notes I have made as an introduction. However, something which I think is still worth mentioning, and Vidmar has made quick reference to that, is the interaction between Mendel and Nagaly. You probably know that, well, all of you know, it's preaching to the choir, that the findings of Mendel went unheeded for almost 4 decades. And there are several reasons for that. One of those reasons might have been, and probably was, that Mendel sent out one of his 40 reprints of his publication in 1866 to Carl von Nagaly. And Nagaly was the prime botanist, to my understanding, in Germany at his time. And Nagaly had a favorite organism or one of his favorite organisms to work with was hawkweed, Hiratium, Habichtkraut in German. And so they decided, I don't know whether it was on Nagaly's request or whether Mendel offered that, to reproduce his findings on pea with hawkweed. And they totally failed. And one of the reasons, or not one, the reasons why they failed to reproduce the pea data in hawkweed is that hawkweed has certain idiosyncrasies in its reproductive system where meiosis is frequently suppressed and clonal seeds are formed. And clonal seed formation commonly is referred to as apomixis. And therefore, today it's obvious it couldn't be reproduced from the beginning because they have been working with the wrong species, or because hawkweed is an exception to the rules. And there are, of course, with each rule we have in life science, there's at least one exception. Well, and that now brings me directly to the first speaker in our session. Because similar to pollen mother cells, to a pollen mother cell switching from the sexual to the apomictive state in the life cycle of a plant, for instance, the differentiation, similar to apomixis, the differentiation of neural stem cells depends on the activity and the orchestration of numerous genes. The mechanisms that lead to organellar diversity in such neural stem cells and the consequences for disease will now be described by Magdalena Götz. She has been a member of Leopoldina since 2008, and she was bestowed with the Mendel Award in 2019. Being the director of stem cell research at the Helmholtz Center in Munich and head of the Department of Physiological Genomics at Ludwig Maximilians University, LMU, that's different from MLU, that's the Martin Luther University here. She is interested in neurogenesis, also in the context of brain injuries. Please welcome her to the podium, and we are looking forward to your lecture. So, the stage is yours.
Thanks a lot. Well, it's of course great to be here again. Actually, the Mendel Medal celebration was not even so long ago. And indeed, rather than talking about genes and even transcription, even in the ages of lots of single-cell transcription, I will take you predominantly to the proteome, and I will give you an example why a ubiquitous expressed protein in all cells of our body actually causes a brain phenotype when mutated. Finally, there is a mutation in a gene. So I'll take that then to the next level and discuss with you how protein diversity of organelles plays a big role. But obviously, I wouldn't be standing here due to our, let's say, unexpected discovery some while ago that in fact the neural stem cells are glial cells and glial in nature. And speaking about unusual discoveries, probably nobody would have believed me. If not, some colleagues then would have jumped on this topic and also found the same. Now, this, of course, changed our view. So this is true for developing neural stem cells, but also adult neural stem cells. And that, of course, changed our view on brain injury and the glial cells. Don't worry about details here. These are different glial cells. Obviously, these ones, after brain injury, they form a scar and cannot generate neurons, while these glial cells look differently, but also they can generate neurons. So obviously, we were interested in not only to understand the mechanisms of neurogenesis, but also how do they compare to these other glial cells. Now then, indeed, we took on a long journey of RNA sequencing, isolating by fluorescent activated FACS sorting these radial glia cells from the developing forebrain, looking at their gene expression, then isolating the adult neural stem cells, their neighboring cells, and in fact their offspring, the neuroblasts, and then isolating the differentiated glial cells, astrocytes for the experts and oligodendrocyte progenitors, be that as it may, to compare their gene expression. Now, I'm showing this cluster diagram, which basically highlights the overall similarities in gene expression between all these different cell types that I alluded you to, as a means of introduction to make you alert of some unexpected similarities again that we found, like why would the adult neural stem cells specifically be similar in transcription to some glial cells from a particular brain region? Well, it turned out that this is the case because, in fact, these glial cells lifelong self-renew and generate further glial cells, which is something very unusual in the adult brain. Let's return to neurogenesis. I alerted you to the fact of the radial glia cells, the stem cells from the developing brain, and when we look, which are, so to say, by genome-wide expression analysis, their closest neighbor, this is the progeny of the adult neural stem cells. The adult neural stem cells itself, they seem to be very different from the neural stem cells in the embryo. And we can, of course, discuss a bit more about why that is the case. For now, I've reached the point in my introduction when we can become more specific, because now we know which populations we should actually look at if we want to identify neurogenic factors, the factors responsible for neurogenesis, because these are adult neuroblast generating neurons, and these are the stem cells in the embryo that generate neurons. And indeed, by the way, just in passing, these are the cells that are transplanted into patients because they are so firmly specified to generate neurons that they can also do that in an adult brain. So obviously now we look at the genes that are selectively expressed in these populations that I outlined to you that generate neurons, and I would like to take you on a brief journey of discovery of some novel players in this area. And this will lead us to the organelle heterogeneity. So first not first, but at some point we picked this alleged 80-hook transcription factor that was in the databases 80-hook transcription factor. Turned out neither to have an 80-hook, nor was it a transcription factor, but actually it was a novel centrosomal protein that mediates the delamination of the neural stem cells from their niche. Now, this was an important finding for us, at least, because it's not only the case for neural stem cells, but also generally for epithelial cells when they delaminate and become more mesenchymal. And this epithelial mesenchymal transition takes place in many cancer conditions and metastasis formation. So that was interesting, but as always, interesting and surprising discovery, it takes you even further, because we realized that this protein that is at the centrosome and is so important for the stem cells leaving the neural stem cell niche regulates obviously this migration to the next position where transit amplifying progenitors are, but then it needs to be down-regulated again for the next migration, meaning the centrosome here has this protein and then must lose it again. And that really made me think a lot about centrosome composition. And generally, obviously, many of us may think the centrosome is quite homogeneous between cells. It must organize, obviously, the cell division and or microtubule organization. So that must be very common. And it's probably due to this concept that, in fact, we had no comprehensive centrosome proteome of any cell from the brain. So obviously, having seen the specificity of a single protein, we wanted to know how is this comprehensively? How different are centrosomes? Well, between organs and even between closely related cells. So what we embarked on was, of course, using human-induced pluripotent stem cells that we can differentiate into neural stem cells and then neurons. The advantage of this culture system, 2D, is that you get pure populations of neural stem cells or then pure population of neurons. So that's very good for comprehensive proteomics. And what we decided to do is a spatial proteomics by actually immune precipitating 10 bait proteins and looking at the interactors, both in the neural stem cells and the neurons. And when we did so, what we found was quite surprising, because in fact, we found a couple of hundred new centrosome interactors that were never seen before at the centrosome. So that must mean that the neural centrosome indeed differs in its composition from previously known centrosomes, hex cells, Drosophila, and many more. Now, even when we looked at the, let's say, functional GO terms, so the significantly enriched groups of protein function, what we found were a lot of RNA splicing, metabolic process, and so forth, besides the known function, of course, of the centrosome in the neural stem cells, like mitotic cycle, or the cilium formation for the neurons. Now, as we did spatial proteomics, we can also see how the localization of these RNA binding protein, and I've highlighted some here because some of you may have heard about Fragile X protein, also an RNA binding protein, which we now found at the centrosome, how these change actually even in position at the centrosome from this localization here, where microtubules are anchored, to different localization in neurons. Now, obviously, we also did a lot of controls to verify that this is specific and has absolutely no correlation to the total abundance of the protein. But let me now turn to the question, does this matter? Now, one way to probe this was to look at mutations, so we do come back to some genes, gene variants that have been found in patients with neurodevelopmental disorders, but as they have been found only in a single family or in a single patient, one does not know if these mutations are causative or not. Now, we wanted to ask the question if we could prioritize such gene mutations by overlapping them with the centrosome proteome, the known one, or in particular for brain disease, so to say, our novel proteome. Now, when we do so, we find common significant overlaps with autism spectrum disorder, for example, but we also find highly specific significant overlap with another disease, only with the neural stem cell centrosome proteome, not the neurons and not the known centrosome data sets. Now, what is this disease? It's abbreviated here PH, which stands for periventricular heterotopia. The picture of this from a patient is shown here, and as the name says, here there is gray matter. Gray matter are the neurons. Here there is gray matter ectopically positioned at the ventricle, periventricular heterotopia. So now we found a significant overlap, fine, but what does that mean? So obviously we picked a candidate, and this candidate is what I was alluding to. It's a ubiquitously expressed protein in any cell of your body. It's an RNA processing factor. And of course, we choose it for good reasons, because several of its interactors were also at the centrosome. And this is the new mutation here that has been found in a patient with such a disease, the periventricular heterotopia. Now, we modeled this disease in the mouse and looked if this mutation would cause any similar phenotype. What you can do, you can inject, so to say, plasmid DNA into the ventricle of a murine embryo and then electroporate, so apply a current to drive this DNA into the developing forebrain. You can see we can do that with control, just GFP, or with a wild-type form of this factor, PRPF6, or the mutant, the periventricular heterotopia mutation. And as you can see, indeed, only in the mutant do we get a significant accumulation of cells there. Mind you, also in the patients, of course, many neurons actually make it to the right positions. Here would be the differentiating layer, and only some get stuck, which is exactly what we found in this. Now, I told you this is a splicing factor, so obviously we wanted to know if splicing changed, and we isolated the cells by fluorescent activated FACS sorting at a time 1 day after in utero electroporation, so 1 day after applying it, when there was no phenotype yet. And sure enough, we found changes in splicing, exon skipping in particular, or intron retention. So then, of course, we looked which genes are misspliced and how could they explain a phenotype at the centrosome. Now, we took as a candidate this kinase because of its substrates, namely it phosphorylates microtubule-associated proteins. And the microtubule organization from the centrosome is important for the delamination of the cells mediated by the protein ACNA, as I briefly alluded to before. So, in a nutshell, what I'm showing you here then is that indeed the correctly spliced BRSK2 isoform can indeed rescue the phenotype. Here you see cells at the ventricle, here also, but not when this mutant PRPF6 is co-electroporated with the correctly spliced form. So, what we have then shown, and this is obviously very difficult to see here at this slide, is that indeed the splicing target of PRPF6, this BRSK2, is also brought to the centrosome as RNA, and we also find the protein there. So we would propose a model where this splicing factor brings its splicing target to the centrosome, it's locally translated, and can then phosphorylate microtubule-associated proteins there that are important for delamination. So this is why a ubiquitous protein, when mutated, only has a phenotype in the brain, because only there it's actually at the centrosome. Now, beyond this very specific case and the surprising general heterogeneity, let me then emphasize that at least for the centrosome, we would say that the cell type specific composition of the centrosome that differs hugely from other cells really matters for development, because cells wouldn't delaminate if that doesn't work properly, but of course also for disease then. Now that was one organelle that I wanted to discuss with you, but in fact we also found other organelles that are commonly considered to be very homogeneous, like the nucleoli, to contain proteins, or actually be surrounded by proteins. This factor here, a novel nuclear factor that we discovered some while ago to be involved in brain folding, this factor regulates phase transition in the nucleus, and in fact regulates nucleolar size due to its assembly outside and function. Namely, it promotes M-phase progression and translation there. So yet another organelle to think about that. But this brings me to the short next chapter that I'd like to discuss with you, namely, how can we use these very general insights from studying the mechanisms of neurogenesis then for neural repair. So now I described to you some new factors that we identified and that brought us to this organelle heterogeneity. How does this actually apply also for repair purposes? Now, as I told you, the original discovery of radial glia cells being neural stem cells in the embryo, but then normal astrocytes or other glial cells losing the neurogenic capacity brought me to the idea some while ago, again, beginning of this century, that we could simply use the fate determinants, transcription factors, expressed in these radial glia cells to then turn these glial cells, these astrocytes that would never generate neurons, into neurons. And indeed, we can do so by using the right fate determinants to turn them into glutamatergic neurons, excitatory neurons, or inhibitory neurons, all borrowing from development. Now, briefly to this factor that I described to you that regulates nucleolar size and function, indeed, this factor is not only important for reprogramming, because when we knock it down, we get fewer neurons, but it can also double reprogramming, but only if it contains the domain, the intrinsic disorder domain, that actually mediates the phase transition. If we delete it, it doesn't work also in reprogramming. So with this, let me briefly also show you that this direct neuronal reprogramming does not only work in vitro, where we can study mechanisms, but that it also works in vivo. And again, let me maybe make the point that this is also really a long-term enterprise to get this to work from the original discovery to then really improvement and high efficiency. So this is a brain injury. It's a traumatic brain injury. And in red, you can see the virally infected proliferating reactive glial cells. And you can see that they don't generate neurons because here it's black, so to say. But when we stick in some developmental fate determinants, thanks a lot, you can see some white cells here. And indeed, these are young neurons. They are very young and immature, so not super useful to repair your brain after injury. And in fact, we had to go back to the drawing board, first in vitro, and for many years trying to identify the main hurdles. And in fact, what we found was that one of the main hurdles that actually kicks the cells into death is the metabolic conversion. And the young neurons that convert from glia into neurons, they die by a special type of cell death, ferroptosis, due to excessive reactive oxygen species. I'm telling this to you because this shows you how important this metabolic conversion is, independent of the neurogenic fate determinant. So we are back to our injury model. We again inject the virus expressing one neurogenic fate determinant 3 days after injury, and then we look 10 days later. And as you can see, the neurogenic fate determinant on its own generates very few neurons. But if we help the cells to fight reactive oxygen species, also by giving a vitamin E analog, then we get 90% cells that convert into neurons. And in fact, these are much more mature neurons now expressing mature neuronal markers, a pyramidal-like morphology, and a specific subtype identity. So with this, I hope I could show you that indeed it works in vivo in the injured brain to turn, in that experiment, it was proliferating glial cells, but we can also convert other astrocytes efficiently with 90% efficiency into the right neurons of this brain region. Now, of course, I told you that, in fact, there is this major hurdle, the metabolic conversion, and that excessive reactive oxygen species are caused in the conversion process. Well, of course, we wanted to know why is this actually the case. And what we found out is that this organelle here, that all of you know is important for metabolism, the mitochondrion, fails to convert properly. Now, what do I mean by this? Again, we are back to organelle heterogeneity, because in fact, the mitochondria of astrocytes and neurons differ by 20% of all their proteins. Now, obviously, when we convert astrocytes into neurons, we need to also convert the mitochondria. But as it turned out, these 20% of neuron-enriched mitochondrial proteins, they are not turned on if we don't help them to do so. So with this, I hope I could convince you that the place where the protein is an important part of its function, and that moving proteins between different organelles or putting them at specific organelles in specific cell types is a regulatory layer to amplify protein functions. And I hope I could convince you that indeed this matters in ontogeny and phylogeny, but also for disease and repair. And with this, I'd like to thank the fantastic people I have the privilege to work with on an everyday basis in the lab, our fantastic collaborators, not the least our proteomic expert, and of course, you for your attention.
Well, thank you very much for this interesting piece of neuroscientific work that you presented to us. And well, for me as a plant geneticist, it was particularly fascinating, and I'm looking forward to seeing questions from the audience. Where are the neuro scientists?
Just a technical question. Your proteome analysis, does it reflect also membrane proteins or only soluble proteins? Because when membrane proteins are not discovered in your technique, you may miss several important factors. Well, I'm thinking of my colleague at the University of Zurich, Martin Schwab, who has discovered NOGO, for example, which is a membrane protein, and I wonder whether it was in your screen.
No, it was not, because we specifically profiled the centrosome, only the centrosome proteins, or only the mitochondrial proteins, avoiding all other proteins in order to know what is specifically at this organelle.
Okay. Well, maybe it'd be interesting to widen this analysis to membrane.
Sure, but if, as we were interested in mitochondrial function, we needed, of course, to profile the mitochondria, and we have found there. As we were interested in centrosome function, we looked at the centrosome. If we will be interested in membranes, that plays many important roles, of course, then we'll go for that. And the interesting point that we are now analyzing is, in fact, how proteins shuttle between these compartments. And there, of course, the membrane is also an important part.
Thank you.
Well, yes, please.
About 20% of your mitochondrial proteins are different. Let's put it this way. Is there some system in there, or is it specific proteins? Are these neural specific proteins which you don't find in liver or something like that? And what is the message out of that? Because they are, I mean, I assume many or all of them are come from nuclear genes, right?
Yeah, so absolutely, as anyway, most mitochondrial proteins are nuclear encoded. These are nuclear encoded. They are of certain groups, but I just couldn't tell you all the data from that. They, of course, have to do with fatty acid metabolism in the astrocytes. They have to do with oxidative phosphorylation in the neurons. But interestingly, there's also common groups like antioxidant proteins. And even some proteins of the same family are astrocyte-enriched or neuron-enriched. And when we activate the neuron-enriched one of the same antioxidant protein family, then we can improve reprogramming, but not when we activate the astrocyte-enriched one. So the message, you asked for the message is that, of course, these proteins help the mitochondria to perform its specific metabolic function in a specific cell context, and therefore they matter for the proper function of the cell. And if we do reprogramming, we must make sure that the mitochondrion also functions well. But so far, nobody had looked at that in reprogramming.
Well, we need to stick to our schedule,
Sure.
and therefore we need to leave it here. Nevertheless, I hope you will still be available to discuss further questions during the coffee break or
Absolutely.
even during the dinner. Therefore, thank you very much.
Thank you.
Indeed. And we will move on to the next speaker, Conrad Sandov, where we still will stay in the field of neurobiology, and we will learn about the molecular basis of neurodegenerative hereditary diseases, or a disease. Conrad Sandov investigates cellular metabolism. He is heading a research group at the University of Bonn and has been elected to Leopoldina already back in 1999 and received his or the Mendel Medal in 2001 for his outstanding research into sphingolipids. Today he will report on his findings on neural gangliocyte, you can express it better than me, gangliocyte degradation. Please welcome Conrad Sandov to the stage.
Dear President, dear Dr. Grana, I'd like to thank you very much for your introduction. Dear ladies and gentlemen, I'm mainly talking on lipids as building blocks and regulators of biological membranes, especially on gangliocytes. The name is since they were discovered in neuronal cells by Ernst Klenk in the late 30s. These are complex glycolipids which are needed for the function of cellular neuronal membranes. Gangliocytes are key components of these ganglion cells and membranes, and their turnover, the destruction, in lysosomal compartment is regulated at the genetic and at the post-translational level. Now, was denn hier das nächste? Okay. Now, when I started with my thesis work at the Max Planck Institute for Psychiatry in Munich, in the research group of Professor Jackiewicz, my thesis work just failed. Since the components I was asked to isolate, I never discovered in the normal human brain. They are still not existing today. So I thought about what to do now. Going back to Professor Lünen at the university, asked for a topic over there, or looking around in the laboratory, what to do. So in a huge refrigerator, which was of wooden material at that time, I found at the back a brain, a human post-mortal brain tissue in acetone, with a diagnosis of amaurotic IUC written on the glass. So I thought that's very interesting. I was just learning and doing thin layer chromatography for separation of lipids, since Jackiewicz developed these methods at that time in the beginning of the 60s last century. Now, amaurotic IUC is an inherited disease, recessively inherited disease. It was known for clinicians at the time, and it's a neurodegenerative disease. You see the neurodegeneration in the macula, at the back of the eye, this cherry red spot. Can you take down the lights a little bit so that one can see someone? Nobody there? Okay, I can't do it. So this cherry red spot at the macula tells us that the neurons are already died, they are degenerated, and you see the blood vessels through. Under the electron microscope, this is a nerve cell here under the electron microscope of a patient, infantile patient of this hemerotic idiocy. You see the cytosol and the axon hillock are just full of storage material. And today we know this storage material are lysosomes, which are storing all the materials they should digest, but they can't do it anymore. So the lysosomes are the stomachs of the cell. They have a huge turnover, and the degradation material they present to the cytosol for new biosynthesis of macromolecules and for energy metabolism. Now, I used my small knowledge on the newly generated form to separate lipids, membrane lipids. I did this on a Saturday. Nobody was in the lab, since I was certainly not allowed to take some material from the refrigerator. So this is a control run, and this is a run from this young patient passed away with 3 years of age. And I found two big components in there are called substance A and B. These are now the main storage compounds in Tay-Sachs disease in the brain. And since these kind of gangliosides occur almost exclusively in neuronal cells, it's a neurodegeneration which occurs by this storage over there. So I did some chemical tests on the nature of these components. I found sugars in there, hydrophobic anchor, what we call now ceramide. And I was lucky that a little bit later, Kuhn and Wiegand from the Max Planck Institute in Heidelberg at that time presented in the literature the first correct ganglioside structure. It's one of the major gangliosides. And using this, I degraded this chemically. I could identify these storage components and separate them. So after I had this and some information, 2 weeks or 3 weeks later, I showed this to my boss, and he was very much surprised to see this and didn't say anything for a while. Then he said, "What are you doing now?" I said, "I'm a chemist, and I will try to isolate the components, to look into the structures, and then to look for the catabolic enzymes which are needed to degrade them and missing in this inherited disease in these patients." Now, the storage material, the extent is given here. The structures I found out by this kind of mechanisms is given here, the GM2 and the GA2 in the first patient you see here. In common, they and later on others have something, which is a terminal sugar is an N-acetylgalactosamine in beta-glycosidic linkage all the way through. So the point was I had to look for a galactosaminidase to split it off. It was not known in the literature at that time. What we knew at that time are glucosaminidases, not galactosaminidases. So since their sugars are very similar, glucose and galactose, I thought I would try them. So I separated the glucosaminidases into the A and B position into 2 isomers and analyzed, then asked for material which was enzymatically active. In 1966, I got material from Harvard University, and I found a complete deficiency of the A, hex A enzyme activity, and the B was very much increased. So I thought that was my first finding, that's it. But it was not. They sent another case, this one down here. So this hex A spot was very much increased, not missing. So this was a contradiction in itself, so I could not publish this. I was waiting for another case I got from Göttingen University, and here I found a similar principle here. It was very mysterious. Then here I found 2 enzyme activities, B and A, both missing. There's a minor one, which is now called hex S, which you don't see in these places up there. So this I wanted to publish. I failed. All the journals said, thank you. One said, it's outside the scope of the journal. And so I presented this at an international meeting. I got a lot of questions. The problem was with the organizers, since the people even asked after the second and third presentation still questions to me. And later on, 2 or 3 years later, the first paper came up which published similar patients. So this was in Coimbra, in Portugal, 1967. Now, the next run, of course, was for the structure of the enzymes and what is going on. What is the reason that if you have these enzyme activities here with synthetic substrates, but also with this GA2 substrate, with the native, with this native at least one, that you still have the storage of GM2. This is the main storage compound. So a look into this situation took several years, and I show you nothing. This one is better than nothing. Okay. So first of all, what is the enzyme structure? These are heteromers. Homodimers, heterodimer, homodimer. This is composed of beta subunits, and the active site is at the interface to split products, substrates to products. This one is a heterodimer, which also has 2 active sites at the interface. And it turned out that one splits the GM2 and the other one not. So they have different substrate specificity. They are coded by different genes. The affected genes are given here. And each of these diseases, Tay-Sachs, has an infantile form, a later onset form, a juvenile, adult form, and so on. I don't go into, I don't go into this. Oh, I'm sorry. Now, here we have only one kind of substrate specificity. Glycosaminoglycans are split. Here and here 2, but different structures. Oligosaccharides are split by these different structures. Glycolipids, but GM2 only by this one. The others have minor or no deficiency at all. So the question now was, how is this regulated and what is going on? So later on, this disease was then where I had 2 enzymes missing, hex A and hex B, due to the common subunit, the beta subunit, now mostly called Sandov disease. This is a standoff now, how the degradation of the main storage compound, GM2, is regulated. This applies similar to other diseases, to other lipids, sphingolipids stored in other diseases and so on. But this is one we have best analyzed so far. Now, we are here within the lysosomal compartment, so in the stomach of the cell. And within this lysosome, there are vesicles, which we call intralysosomal luminal vesicles. And these vesicles you see here, the lysosome would be as big as this one. It's an organelle. We just heard about organelles, but this is a picture for this. It's outside the screen. Now these lipids, these are all sphingolipids, they are not water soluble. So we don't have the normal theory. They are bound to membranes. They are at the surface of the membrane, and especially they concentrate for degradation on the surface of these intraluminal vesicles. And the proteins involved in the degradation, the hexosaminidase A with alpha beta, and with this lipid binding protein which I didn't tell you here. We looked why, in this one case, we had high enzyme activities and still the storage. We found a lipid binding protein coded here. We call it activator protein, a liftase, which is able to bind to the substrate on the membrane and to lift it up and to create a Michaelis-Menten substrate complex on the surface of this membrane. And the speciality of this membrane is that it doesn't have a glycocalyx, no glycocalyx. So glycocalyx is rich in mucopolysaccharides, glycosaminoglycans, and other sugar stuff polymers on all our cells, especially on the surface of neuronal plasma membranes, but also on the perimeter membrane of the lysosome, I mean inside. In order to protect this perimeter membrane of the lysosome for degradation, against degradation. Otherwise, you wouldn't sit here. That would be a very fast process. So they form this complex with the lipid, and we identified binding sites between the activator and the enzyme and the substrate, the activator with the substrate, and all together to this complex. And here is the sugar is released, and the rest of the GM2 is still sticks to the membrane, is now a smaller ganglioside called ganglioside GM3. This is a situation in the model experiments what we have in vivo, what works in our brain, since these kind of gangliosides specifically are in neuronal membranes. Das geht nicht. Now the point is that these enzymes are also active against synthetic substrates. In medical sciences, these medical substrates, water soluble, are mainly used to identify enzyme defects, to identify the activity of these hydrolases and so on. Now in this case, the substrate is called MUX. This is 4-methyl-amylopyryl N-acetyl beta-D glucosaminide 6-sulfate. So it's anionic like the GM2. It has the linkage. The structure is not here. It was not copied. I had the structure here for this data, and here the glycolipid structure as natural substrate radio-labeled to measure the degradation by the enzyme. If we do this, if we just mix the enzyme with the substrate, here with the water soluble, we get full activity. And on all the other conditions here, the same here, always about the same, only small differences, very small differences. So it's a very good method to measure in a patient's sample, tissue sample, blood sample, how much of the activity is available. But this does not tell how much ganglioside, lipid splitting activity is available. Since if you do the same mixing between enzyme and substrate down here with this substrate at the membrane surface, it's 0. Exactly 0.0. Very sensitive assay. If you add now here the activator protein, you get a little activity. If you add in addition anionic lipids like BMP and the activator, you get nice activity. We had for decades the same one. Then we studied many more lipids, building blocks of these intralysosomal vesicles, anionic, and others. And if we add thiamide, just the hydrophobic anchor here as a free molecule, then we get a real physiological relevant activity for the degradation of this ganglioside stored in Tay-Sachs disease. This is the main component which makes this neurodegeneration. Now the point is that another lipid like sphingomyelin, for example, inhibits. If, however, we degrade sphingomyelin with acid sphingomyelinase, then we convert it to thiamide. So we get come from an inhibitor to a stimulator, to an activator by the lipid, not by a protein, not by a gene or so, but just by a membrane lipid. And this is a physiological relevant activity we would expect. Now this is another example now for post-translational modifiers, anionic lipids which may enhance the degradation of another ganglioside, the GM1, which was first characterized by Kuhn and Wiegand, as I mentioned, by the beta-galactosidase. This disease is now called GM1 gangliosidosis, and it was basically, first description was my thesis work after what I said you before. And if you look into these data, this is a rate of hydrolysis you have here. If you have neutral membranes, no anionic lipids, and no activator, these are the red bars all over here. It's very little. It's not enough for staying healthy. If we add one of these lipid binding proteins, the GM2 activator protein blue, as I mentioned before, or the saposin B, another one discovered by Jackiewicz, you see here green, both stimulate in this case. So we have 3 genetic modifiers in this situation, the enzyme and 2 different activator proteins. If we, however, include anionic lipids in these substrate carrying membranes, then we get much more hydrolysis. It's 10 to 50-fold about these red bars. So it's really meaningful. It's a lot of regulation. Now I'm coming to the end. I will show you only this overview here on secondary post-translational lipid storage we have in these inherited diseases. These are some factors I don't discuss further which stimulate. For the diseases, this is more important to tell you lipids in these membranes which inhibit. So that will be not a primary storage compound then organized by a gene defect, but a secondary accumulation. So the sphingosine bases, sphingosine and sphinganine, we found out by biochemical studies, they inhibit the catabolism of these lipids I already showed you, for example, in Niemann-Pick disease type C. So there is no defect in the further handling of these lipid backbones in this disease, besides the inhibition of their catabolic proteins by these primary storage compounds. And it's a real inhibition. Another example is that the chondroitin 6 sulfate. This is one of the primary storage compounds in 4 different mucopolysaccharidoses. These are also inherited diseases where glycosaminoglycans are coming up, stored, and they inhibit also the GM2 catabolism in all of these 4 mucopolysaccharidoses. Or the sphingomyelin, which I already mentioned before, is a major lipid in the neuronal plasma membrane to stabilize the membrane, but when it moves to the lysosome for degradation and is not degraded, while the degrading enzyme acid sphingomyelinase is deficient, for example, in Niemann-Pick diseases A and B, cholesterol is accumulating tremendously, despite no protein is defect to remove cholesterol from the lysosome. It accumulates on a molar level as high as a primary storage compound. It's really amazing. What is going on? Sphingomyelin inhibits the cholesterol transfer protein, NPC2, which is one factor to get cholesterol out of the many others, out of the lysosome to reach the ER for regulation, or the mitochondria for the biosynthesis of all these steroid hormones. So this we see in Niemann-Pick disease type A and B. This really comes up. It also inhibits the hydrolysis of these lipids in 3 different other diseases we studied, and the solubilization of membrane lipids by the lipid binding proteins. So far, we have 6 or 7. I talk only mainly on one of them at the moment. Also, cholesterol itself is an inhibitor of the hydrolysis of GM2 in Niemann-Pick disease type C. It inhibits the solubilization of GM2 by these activator proteins to transfer to other membranes within the cell or in model membranes. So in summary, I would like to say that genetic and post-translational modifiers generate cascades of pathology. And the point is that not only genes and proteins are involved, but in this case also lipids. And the brain we have is the lipid-richest organ besides fat lipid. But these are triglycerides. I'm here talking on sphingolipids and other membrane lipids. Thank you very much for your attention.
Well, thank you very much for providing us interesting insight how genetics translates into biochemistry and into cell metabolism at the end of the day. And I open your presentation now for discussion. And perhaps to break the ice, I was wondering whether your findings are leading to curation of the disease or the 3 diseases you described, or whether at least we can or you can achieve a treatment.
There are a lot of attempts to achieve a treatment, but the treatment is only transiently. They cannot prevent the disease, they cannot stop it, and the kids or the adults will eventually die. But what is now going on is the intraceutical. So to use pipettes to pipette within a virus a gene of a missing enzyme, for example, a gene into the ventricles of the brain, and to get it into the neuronal cells. And this has all been studied in animal samples of these diseases. Animal models are available, and in animal models it is working. So you really generate the biosynthesis of the missing hydrolase within the neuronal cells by this procedure, and they reduce the storage and prevent it. In humans, there are experiments at the experimental levels running at Harvard, at MIT, in Southern California, and in Kingston, in Canada. No results so far known to me. Some called me up, discussed problems, and so on. The problems are enormously. The way has to be done this way, since you cannot inject these therapeutic agents just in the blood. You don't get it through the blood-brain barrier. So therefore, they go directly into the ventricles.
Are they applying liposomes, or are you injecting RNA directly?
They can use liposomes, they can absorb, or they can, something like liposomes. That's the only way.
Okay. All right. Any questions from the audience?
No.
If this is not the case, then thank you very much again, and we will certainly continue the discussion over coffee or dinner. Well, let's move to the third talk in this session. And this talk will address a very fundamental question in biology, namely the differentiation between self and non-self, which is one of the fundamental challenges at least to my understanding, in any living system. Just think about the increasing occurrence of autoimmune diseases in humans, which are the result of a failed differentiation between self and non-self. In plants, the interaction between a pathogen and its host, also fairly complex, admittedly, mostly follows Mendelian rules of inheritance. Our next speaker, Regine Carmen, will introduce us to this exciting topic by describing the interaction of the ongoing battle between a smut fungus named Ustilago maidis and its host plant, corn or maize, evidently. Regine Carmen has spearheaded the field of molecular phytopathology in plants. She headed the Department of Organismic Interaction at the Max Planck Institute in Marburg for 20 years. Being a member of the Leopoldina since 2008, she received the Mendel Medal in 2011 for her groundbreaking achievements in phage genetics, which she probably will not address today, and developmental biology and developmental genetics of Ustilago maidis, this nasty pathogen of corn. Please welcome Regine Carmen to the podium.
Thanks, Andreas, for the kind introduction. And so what I want to do today is take you back 40 years. And 40 years ago, I basically decided to switch my research subjects and leave site-specific recombination, which I was studying in phages, and move on to a new system where I was basically fascinated by the genetics. And it's a fungus that infects corn and causes this ugly-looking tumor-like structures, here mostly shown in the cobs. And if you see closely, you see there is some black dust coming out. And this black dust are the fungal spores. And this really gives the disease its name. It's called corn smut disease, because the corn plants, the infected corn plants, have a smutted appearance. So this is a particularly nice example of an infected cob, and where you still see some kernels which are uninfected, and you see the results of the infection, these blown up kernels, tumor-like structures, and they are filled, you see the black spots here, with fungal spores. Now, what is fascinating about this system is the biology. So, fortunately, we can grow the fungus in its haploid form. It grows like yeast. However, if you take a pure culture and inject it into corn, there is no disease whatsoever, no symptoms. So to get from this non-infectious form to the one able to infect corn, you need to have mating of two compatible strains that differ in their mating types. They fuse and there's then a dramatic morphological transition. You get a true filament which shows tip growth, and it's a dikaryotic filament. Now, on the leaf surface, this dikaryotic filament can differentiate, form an infection structure. You get infection, and then there is massive proliferation initially as a dikaryon, and later as a diploid cell inside the plant. And then you get spore formation, they are diploid, they can germinate, undergo meiosis, and the whole thing starts all over. Now, the fascination about this system is that infection by the plant and ability to produce spores, that is, the end sexual development of the fungus, are intimately coupled. So there is no disease if you do not have sexual development. So and I found this incredibly attractive because it meant, well, that's at least what we believed at the time. If you understand sexual development, you understand how this fungus becomes a pathogen and is able to infect corn. So, fortunately, 40 years ago, there was some wonderful genetics already done on the mating system of this fungus, which allows cells to discriminate self and non-self. And it's basically all about what Mendel called inheritable genetic, he didn't use the term, genetic elements. So it was clear in this system that compatibility, that is the ability to fuse and form tumors, infect plants, is regulated by two loci, which are designated A and B. So if you have a haploid cell, it has one particular A locus, and the A locus exists in two alleles. And it has one particular allele of the B locus, and the B locus exists in many alleles. So haploid cells are unable to infect plants. However, if you combine haploid cells, there are combinations which on plates, so these are real crosses on plates, show filamentous growth. And when you inject these combinations into plants, you get disease. So this means, and these other combinations fail to switch to filamentous growth and fail to cause disease. So in order to cause disease in this system, you need to be different in A, and you need to be different in B. And once that is assured, the cells can mate and the cells can cause disease. So in addition, it's possible by genetic tricks to isolate diploid strains. And these diploid strains can be heterozygous at A and homozygous at B. Then they are unable to infect plants. They can be heterozygous at B and heterozygous at A and B. Then they are filamentous and can infect plants, cause tumors. And if you have a strain, a diploid strain, which is homozygous at A and heterozygous at B, this strain is also able to cause tumor formation. And the conclusion from this result is that it's really heterozygosity at the B locus, which is necessary for pathogenic development. So the A locus seems to be essential for cell fusion, but if you bypass the step of cell fusion because you are working already with a diploid, you don't need a heterozygous A locus anymore. So this meant 40 years ago, to understand this system and to learn what is causing this, what is allowing this fungus to cause disease, you need to clone the B locus. And the genetics had actually predicted how it would work. So you start out with a diploid, which is heterozygous at A, which is homozygous at B. It's non-filamentous. And then you transform it with a gene library from a B2 strain. And then this strain should become filamentous, and it should cause disease. Perfect, easy, forward genetics. Unfortunately, I was not the only one having this idea, and it was actually, I was also not the first one who published it. So the group of Sally Leon and helped with a postdoc Jim Kronstadt published this paper in '89, where they had cloned two alleles of the B locus. At that point, you can either give up and say, I'm dropping this system, or you can continue. And I decided to go on, and it took us another year to actually clone the locus and find out what these inheritable B elements are doing. So what we learned from comparing four different alleles of the B genes is that the products have variable domains and constant domains, and these domains are separated by a homeodomain motif. This is not the typical homeodomain, it's only very remotely related to the homeodomain, But finding this motif allowed us to predict that the B proteins are regulatory proteins. And this allowed us to come up with a model. And the model at the time was that the variable regions of these B proteins govern association and activity, maybe, or the constant regions govern association and the variable regions then determine the activity. So this was very fortunate that we could at least propose a model, and this allowed us to publish this paper. And we were very happy because this was my first Ostilago paper. Unfortunately, this was only the start. And the next experiment was an attempt to actually prove by genetics that the B genes have this essential role in pathogenesis. So this was prior to PCR, so we had to try to delete the gene and replace the endogenous gene. And we did that, and this is an original figure, hand-colored. And what we realized is, so if we delete, for example, the B2 gene, pretty much only the open reading frame, this strain was still mating as if it had the B2 gene. So you cannot probably imagine how shocking this result was. Fortunately, we had also deleted the B1 gene, but in this case, we had deleted some of the upstream region. And this strain was unable to mate. And we had a third strain where we had deleted the B2 gene, but we had replaced this part by B1 DNA. And this strain, all by a sudden, now mated as if it was B1. And this meant that there must be a second gene. And being in Berlin at the time, we decided to call these genes B East. That is the initially identified B gene, and B West, that is the newly found B gene. And it's very rewarding that it later turned out that it's really the association of the two B proteins which govern activity. So, and this name, B East and B West, did stick. So we have the situation that we have two divergently transcribed genes, B East and B West. And so, for example, in B1, you get the products of these two genes. In B2, you get the products of these two genes. And the proteins can form heterodimers, but they can only form heterodimers if they derive from different alleles. So you can get an active heterodimer between East 1 and West 2, and you can get an active heterodimer between East 2 and West 1. So proteins from the same allele simply do not dimerize. This tells you why a haploid is not able to infect and why you need the diploid or the dikaryotic cell having two different alleles of the B locus. So self-non-self recognition is really determined by association or non-association of proteins. So having this, it was then very easy to know what has to be done next, because we believed that all we need to do is find the targets of the B genes. So since both genes, B East and B West, contain homeodomains, you get this complex of East and West. The idea was you need to identify the binding sites, and then you know which genes are needed for pathogenic development. This work was largely driven by Jörg Kemper, and he actually established that there is a regulatory cascade where you have very few binding sites, and among the genes which are regulated are again transcription factors which then regulate another series of transcription factors, and so on and so forth. So unfortunately, many of the genes in this regulatory cascade, when deleted, turn out not to be essential for virulence. That was really disappointing and made us wonder what this cascade is doing or what is missing. And what we really missed out considering is that this system is a system where the fungus interacts with the plant. And what we missed out in doing this type of experiments by expressing the East-West complex and then doing chip analysis is that we didn't have the plant. So we missed out on having the contribution of the plant. So what is the contribution of the plant and what is actually happening when the fungus infects corn? Here you see the fungus on the leaf surface in red, and it's infecting a plant which has a green plasma membrane. And you see that upon penetration, the plasma membrane invaginates. Here you see the fungus inside the epidermis, and it's completely encased by the plant plasma membrane. So this fungus is inside the host, but it's never in contact with the host cytosol. There is this membrane in between. And this membrane really creates an extended interaction zone. And it's this interaction zone where the two organisms interact. There is another aspect which I have not told you. Ostilagomedis is a biotrophic fungus. And a biotrophic fungus does not kill its host. Rather, it establishes an intimate relationship where it's absolutely essential that the host cell is kept alive, because only then is it assured that the fungus is supplied with nutrients. So if you have a filament, the filamentous form of the fungus, and try to infect dead tissue, it does not work. So what actually happens when Ostilago gets in contact with the plant? It's recognized, like all microbes that get in contact with plants, and it's recognized via pathogen-associated molecular pattern, or PAMPs. And in fungi, it's usually, so chitin is such a pattern. And this recognition triggers very, very strong local defense responses. And usually these defense responses are so strong that they arrest the invader. So if a fungus is successful, these plant defense responses need to be suppressed. And in this system, this suppression occurs via secreted so-called effector proteins. And these effector proteins can reside in two locations, either in the apoplast, where they usually inhibit plant defense enzymes, or they can be taken up by plant cells and actually modulate plant processes inside plant cells. And one of the big mysteries in fungal plant pathogens is the question how these effectors are translocated. So we know that several Ostilago effectors are translocated, but we have no idea how this occurs. So if you look at the effector repertoire of Ostilago, there's almost 500 proteins which are predicted to be secreted. And the amazing feature is that almost 50% of these proteins are completely novel. So they do not contain recognizable domains. Nevertheless, they contribute to virulence, but usually they have only a small virulence contribution. And for all the effectors which have been analyzed in detail, it has been shown that they interact with plant proteins and they modulate the activity of plant proteins. And what we also knew is that there is a whole set of effectors which are induced during this stage when biotrophic development is established. That is about 2 days after infection. And in a systematic approach, we have concentrated on this group of effectors and have come across something we did not anticipate, and that is 3 genes where individual deletions of these effectors completely abrogate virulence. So we call these genes stop genes. So stop 1, stop 2, and stop 3 mutants do not show any disease symptoms except for some chlorotic spots. That was very surprising because usually, as I told you, effectors do not show a dramatic contribution to virulence. So here we obviously have essential effectors. So what are these effectors doing? To study that, we epitope tagged them, let them deliver by the fungus infected plants, and then made extracts and did co-IPs to find out the plant proteins these effectors are associated with. To our big surprise, did we not get plant proteins, but instead, for example, let's just take stop 2. We get stop 2, but we also get stop 1 and stop 3, the other 2 effectors which gave us the similar phenotype when the genes were deleted. Plus, we got 4 additional fungal proteins, which obviously all proteins were residing in a complex. So we have a protein complex that contains these 3 essential effectors. It's a protein complex of 7 Ustilagomedes proteins. If you look at the proteins, they're completely unrelated. They are all novel. They're very different in length, and 2 of them actually have predicted transmembrane domains, and we have shown by biochemical analysis that these proteins reside in the fungal plasma membrane. So what happens when you delete now the other 4 genes where the proteins reside in the complex? This is a geneticist's dream, and it's really that you get the same phenotype as when you delete stop 1, stop 2, and stop 3. Virulence is completely abolished. So we have 7 genes now with an essential virulence contribution. So what actually happens when you delete any of these complex members? Here on the left, you see the wild type 1 day after infection. This is the infection structure, and the wild type has penetrated and is happily proliferating inside the plant tissue. If you do the same experiment with any of the 7 complex member mutants, you see that they also form infection structures. They enter, but they never get any further than the epidermis. They are stuck, they are stopped in the epidermis, and this is why we call the genes the stop genes, and we have stop mutants, and we talk about the stop complex. So if you look at a later stage, you see the wild type in green here, heavily proliferating inside the plant tissue, and the mutant is basically stuck in the epidermis and is unable to continue growth. Why is it unable to continue growth? The wild type is after 1 day, and here we have stained the surrounding plant plasma membrane in red. You see the fungal hyphae here, surrounded by the plant plasma membrane. And this also exists after day 2 of infection. If you do the same experiment with any of the stop mutants, you can see that after 1 day, it is a bit difficult to recognize. The hyphae is still surrounded by the plant plasma membrane, but at a later stage, there is something very dramatic happening here, and that is the host cell that is invaded by the mutant is collapsing. The plant plasma membrane disintegrates. You see vesicles, and this indicates there is an induction of plant cell death. And plant cell death is really the end for biotrophic fungus that relies on living plant tissue. So obviously, these mutants are unable to downregulate plant defense responses. And I told you in the beginning, plant defense responses are, the downregulation occurs by secreted and translocated effectors. So where do the complex proteins reside? Here you see the localization of an apoplastic effector in a biotrophic hyphae inside the plant, so in red, and you see it's evenly distributed around the hyphae. If you do the same experiment with any of the complex members, you find the proteins, the stop proteins, in these speckles. And these speckles sit on the surface of the hyphae. If you do immunogold labeling, you actually see where the complex members reside. And they reside in structures. And these structures emanate from the fungus, and they extend into the host cell. And here you almost have the feeling that this is a tube extending into the host cell. Sometimes these structures are associated with plant plasma membrane, and by altering the extraction conditions, we have actually shown that, yes, the stop proteins contact pore-forming plant plasma membrane proteins. So based on this finding, and based on the fact that we can at least partially, maybe fully reconstitute the complex, so usually all 7 complex members are only expressed after colonization. But when you hook all 7 genes up to constitutive promoters, and then look by SEM on hyphae. So let's first have a look at the controls, where the 7 genes are not expressed. You see some structures on the surface. But here, when you express all 7 genes, you see that there is a new type of structure sitting on the surface of the hyphae. And we have actually shown that in these structures we find the complex members. So we believe that we have reconstituted the complex, which should aid further characterization of the complex. And with this, I'm actually at the end, and let me present you my model. So we have identified a fungal protein complex that sits in speckles on the surface of biotrophic hyphae. It's anchored via 2 proteins in the fungal plant plasma membrane. It contacts channel-forming plant proteins sitting in the plant plasma membrane. And we believe that it could be a pore, a channel, or a pillows-like structure for effector delivery to the host. The reason why we believe that it's effector delivery is that whenever this structure is missing, the fungus is unable to establish a biotrophic interaction which relies on the transfer of effectors to the host. So if all of this is true, then we have hopefully identified, no, not hopefully, identified the first fungus structure involved with an essential virulence function that is involved in effector delivery to the host. There is a lot known about such structures in pathogenic bacteria, but so far we did not have related structures in fungi. So in the future, we will have to get the structure of this complex. And I'm very happy that Gerd Bange in Marburg is actually planning to work on, has actually started working on this. And I would like to give my thanks to the people who worked on this project, and it took a long time. There were many people involved, and this is the entire group. And in particular, I want to mention 2 people, Nicole Ludwig and Stefanie Reismann, because they, over a period of, I think, 7 years, brought the project to the stage where it could be published. And I thank you for your attention. No, I want to mention, sorry, our collaborators. Timo Glatter, who runs the MS facility at my institute, and the two people, Karen Snetzeler and Karl-Heinz Rexer, who did the EM work. Thank you so much.
Well, thank you so much for giving us insight into this, on one hand, very complex but also instructive system of host-pathogen interaction. Questions, Ulla?
Yeah. Beautiful talk. Congratulations to this enormous progress that you made. I wonder if the 7 proteins, if you try to express them and then put them together in vitro, do they self-assemble into a structure?
Yeah, I would love to know that. I cannot answer that because we have not been able to produce all 7 proteins. And it's not going to be easy because some of them are post-translationally modified, so you cannot just express them in E. coli. Yeah.
Yeah, fascinating talk. So I was wondering, you mentioned that the pathogen also has an interest not to kill the host.
Yeah.
And it's basically preventing cell death.
Yeah.
But you also show that, well, when you have mutants of one of those 7,
yeah.
then you get cell death.
Yeah.
But why do you get cell death if it's not invasive? Why does the plant bother at all?
Okay, so I showed you this one schematic slide. When the fungus sits on the surface, it's recognized. And this recognition is usually so strong that it prevents penetration. So if the fungus gets in, as the mutants do, and do not manage to downregulate these defense responses, then you get the plant cell is killed. It's local cell death, and this then prevents further proliferation of the fungus that relies on living plant cells. Okay, so the plant really induces this local defense response, which is sufficient to arrest the biotrophic fungus, which 200% depends on the living plant and cannot continue growth as a dikaryotic hyphae on dead plant tissue.
Yes, please.
A wonderful story, Regine. This system reminds me of the bacterial type 3 secretion system, for example, but you said, if I understand correctly, there is no genetic similarity between these systems. So is this an example of convergent evolution here?
I don't know. I mean, so if you look at type 3 secretion systems and other bacterial delivery systems, they are much more complex. So it's not just 7 proteins. Honestly, this is why I was careful in this reconstitution experiments. Yes, we see structures, but we do not really know whether these structures are active. Okay, so it could very well be that we are missing additional compounds, because if this is a delivery system, it's going to require energy. And we have no idea so far where the energy is coming from, because all the 7 proteins we have, except for the 2 which have transmembrane domains, there is no indication that there is some kind of motor or whatever that could drive the activity. The other aspect is that, to be very honest, we have no idea in which form effectors are delivered to the host. So is it that they are delivered as proteins? Is it that they are delivered as RNA? Or is it that they are delivered in vesicles? And right now, I tend to speculate that the delivery occurs in vesicles, and that this structure is actually a device that connects the fungus through, I mean, this is non-trivial, through the fungal cell wall to the host plasma membrane, and it could be delivery of vesicles. And this needs to be studied. And we have not, we ran out of time. We have not addressed this yet.
Is a point that also confused me a bit, because originally these proteins were discovered as secreted proteins.
Yeah.
Did I understand correctly?
Yeah.
Yeah. So somehow they must be reinserted in the membrane.
Yes. Well, two of them are membrane proteins. Okay, so they were not discovered in the secretome. Okay, the other 5 are predicted to be secreted, and for one of them, it has actually been shown by the group of Gunther Döhlemann that it is an inhibitor of an apoplastic plant peroxidase. But this plant peroxidase is expressed in a very early time point, and it's only transiently expressed. And we have actually been able to separate the domains in this effector that are necessary for inhibiting this protein, this plant protein, and the function in the complex. So it could be that the other effectors have also additional functions. We have not studied that, so the idea would be that these proteins are secreted, either conventionally secreted. 5 of these proteins are conventionally secreted, and then assemble on these transmembrane proteins. And Gerd Bange is actually trying to reconstitute now, starting off with the transmembrane proteins and trying to find what is the next protein that interacts, and so on and so forth.
Okay, final question by Herbert Leclerc.
This was a little bit my second question. My first question is, you identified 250 new genes. New genes means, as I understood you, there is no protein known so far with a function so that you cannot correlate them with function. But do they have similarities, sequence similarities, among themselves? Question number 1. Question number 2, I have not understood complex. Complex simply means there are interactions among the proteins. Have you shown that?
We have shown interactions between individual proteins. We have shown, we actually went to yeast and have partially reconstituted different subcomplexes, so we can show that there is direct interaction of these proteins, yes.
And this is confusing me because they are somehow in the cytoplasm and they need to go to the membrane, but they meet in the cytoplasm, right? Or are the whole complexes just as a complex going there? And where is the ATP there? Because you need the energy.
Yeah, I very much agree. So we do not know where the ATP is coming from. Where do these proteins interact? I would assume that you start out with the proteins residing in the membrane, and that you add in the apoplast to this structure sitting in the membrane. But this has not been building up. This has not been proven. So we have been looking. First answering your first question. These proteins are completely unrelated among themselves. There is no relation, but they are fortunately, they are interestingly at least found in all related fungi. Okay, and we have actually used the feature that this complex is essential for virulence to set up a screen in yeast with the screening center in Dortmund. And we have identified compounds which disrupt the complex in yeast, and they inhibit Ostilago infection. And beautifully, they do not just inhibit Ostilago infection, but they also inhibit rust fungi. Rust fungi are also basidiomycetes like Ostilago, but we do not find by sequence the complex there. Okay? So I believe the complex, the structure is more widespread, but it can diverge, and it will be a challenge to get to similar structures in rust fungi. So I don't think it's an Ostilago Medes specific structure.
Okay, we have reached the end of a very, very interesting session. And now, before breaking for coffee, please give a big hand to our speakers. Thank you very much indeed again. They have given us a flavor of how the genetic groundwork that was laid out by Gregor Mendel has been advanced in recent decades. Now, before breaking for coffee, two remarks. Number one is that a speaker of the upcoming session 3, Peter Hegemann, was not able to come here and was not able to deliver his talk online, and therefore we just have 2 talks after the coffee break, and therefore despite running over time, we still have sufficient time for 25 minutes coffee break. That means we are going to reconvene here at 5:25. So thank you for joining in and have a good coffee break.
It's like in a lecture hall. Welcome back after our coffee break. My name is Ulla Bonas and I will chair the next session. And as you already heard in the last session by Andreas Graner, Peter Hegemann was unable to come or deliver a video that we could show, so that slot will remain empty, and we have now the talks of Stefan Mundlos and then a video by Jane Goodall. I'm very much looking forward to this next session, and before our speaker comes to stage, I would like to briefly introduce him. Stefan Mundlos is at the Charité in Berlin and is a member of the Leopoldina since 2004. He got the Mendel Award in 2021, so only recently, last year, and I remember the nice ceremony. He was awarded with the Mendel Medal for his accomplishments in the field of gene regulation and the genetic causes of abnormal skeletal development. And today he will talk about from Mendel's laws to 3D genomics, how sequence determines function and phenotype. And I'm looking forward to your talk. There you are, right in time.
So, thanks very much. Dear President, ladies and gentlemen, it's a pleasure to talk to you. I titled so I made this a little undertitle because I thought in this sort of Mendel setting, I also make this a bit historical and called this a short history of human genetics. And of course, it's a very private view and my personal view, and also, of course, about some things that are related to me and my career in the past. So we talked about Mendel and how long it took until his discoveries really were recognized. And in human genetics, this really took a long time because I think at that time, no one really wanted to accept that humans followed the same rules as peas, which of course is really a challenge. And interestingly, this condition here, which is a brachydactyly, this is very short digits because there are bones missing in the digit. That was one of the first disease that was recognized as being inherited in a dominant fashion. And it's a gigantic pedigree, and this pedigree was actually reanalyzed a couple of years ago, and they finally found the mutation. So it was the first example of a dominant inheritance, and that's what William Bates presented in his lecture in 1906, and compared this to inheritance in sheep and plants. And that's, of course, pretty sensational at the time, is that one would claim that this follows the same rules. And of course it does. And what he said there is that that if we would recognize this, then we would see that the examples and the number of these diseases would increase massively. And of course, that is what happened. So for me personally, the whole story really started when I was doing my pediatric training on the ward. And I have to admit that at that time, my experience in genetics was pretty miserable to non-existent. And what I encountered there was a child what is called intractable diarrhea. So that's a diarrhea that cannot be stopped. So meaning this newborn, you could feed this little girl whatever you wanted, and everything went right through, which of course is a life-threatening condition, at least at that time. So what I then did is that I said, well, it's pretty impossible that she cannot take up any nutrient. So we tested each one of these individually, and for fat, it went all right. So she didn't get diarrhea. Protein also went all right. But in lactose, she got massive diarrhea. The same is with saccharose, and the same with glucose, and the same with galactose. But interestingly, fructose, there was no reaction. So that made the diagnosis, and that's a condition super rare called glucose-galactose malabsorption, and it's a recessive trait. And at the time then, we were able to biopsy, take a little piece of tissue from the gut, and we sent this to a group in the US, and they amazingly, at the time, managed to do something as exotic as an RT-PCR, and then amplified at least a little piece of this glucose transporter and really found the mutation, which was pretty sensational. And it just tells you and told me that early diagnosis can save lives. And also, very interestingly, I think, is that now based on this discovery, there are certain glucose absorption inhibitors developed that are now used as a treatment of diabetes. So that was sort of the first encounter. And then I got really interested in genetics and skeletal disease, and here we basically used the basic things of Mendelian rules, map dominant conditions like this one here, very strange conditions where you have a skull defect, you have missing clavicles, and you have supernumerary teeth. We found the gene, but the other interesting thing then is that this gene, when knocked out in the mouse, these mice have absolutely no bone, which you can see here because of the missing red color. So it was one of the first genes to be essential for bone formation. So this is just an example of many, but what then happened over all these years is that thousands of these disease genes were discovered. Now we are up to around 4,000 of about 6,000 diseases. And that was really a major breakthrough, of course, in human genetics. So how was that possible? The one thing, of course, with the Human Genome Project, this was in 2000, Bill Clinton, in his young age, announcing the Human Genome Project. And the other thing was the development of new sequencing technologies, next generation sequencing, that's what it's called, that changed the whole sequencing massively. And now we are at a price for the whole genome of a human from 1 billion, when the whole genome, the Human Genome Project was ended, to now from 1 billion to less than 1,000 euros. And that, of course, is really changing human genetics altogether. Okay, but then where are we now? At the time when the Human Genome Project was announced, I think it was kind of felt that now all problems are solved, right? So this is just from the time, so okay, we have a look into the instruction book of life, but in the end we have to say we can have a look in the book, but we can't read it. And President Clinton said that's one right thing that he says, there's a lot of hard work that needs to be done. But a lot of people thought, now that the Genome Project is concluded, what else are we going to do? And it was compared to the Apollo moon landing at the time, and I think in a way that's true. And if we now look at what happened, so let's see where we landed, okay? So if we compare that to the moon, then we know we have our genome, we have around 20,000 genes. The entire sequence is known. Now we also have the entire sequence of chromosome from telomere to telomere. Numerous other species have been sequenced. As I said, more than 4,000 disease genes has been discovered, and this is now used by us and many others as a diagnostic tool to sequence genomes or exomes for a diagnostic purpose. So you could think, fine, everything is basically solved, right? But of course, like always in science and life, that's not the case. Because if we look at our patients that we sequence, then we can make a diagnosis with these whole genomes and exomes in roughly about a third. That's already fantastic, but that means that 2/3 remain without a diagnosis in spite of the fact that one thinks this must be genetic, for example, in sick children. So, and that brings me to our major sort of scientific focus at the moment, because what I wanted to show here is that what we've been looking at, and you see here that also in all talks, everyone looks at genes. But this is only 1.5% of the entire genome. So almost 99% is doing something else. And no one really knows what it does. Of course, there are ideas. It's related to gene regulation. So we have millions of enhancers. The majority, actually, of the genome is transcribed. So there are RNA transcripts all over the place where no one knows what they do. For example, there are about 30 to 60,000 linker RNAs without really known function. So I think this is the big challenge for genetics in the future. What does this dark side of the genome actually do? And can we try to understand it? And we can read the sequence here. That's easy, right? Because we have the triplet code. We can translate it and read it and even predict the protein, even the 3D of the protein. But here, the sequence we cannot read. There's absolutely no way. We don't know the code. So I like to look at this as these 20,000 genes as a toolbox. Okay? You have a toolbox, and you have that toolbox in every nucleus. But the really exciting thing is, now how do you use these 20,000 genes? And how do you assemble an organism from this? And this toolbox, interestingly, as you might know, has about the same size in a little worm, Caenorhabditis elegans. Drosophila has a few less genes, but all species more or less have around the same number of genes. So it's how they're used, and that makes the human phenotypes. And that makes evolution, I think. It's the gene regulation, okay? And that makes it exciting, and I think there's a long way to go until we really solve this. So one aspect of gene regulation that we've been looking at is 3D genomics. So what does that mean? In each cell you have, if you take the DNA together, it's almost 2 meters. So you have to pack this gigantic amount of DNA in an ordered way inside each nucleus. And I think one of the major discoveries, in my opinion, is that at least in part, this can now be measured and analyzed and reconstructed. So what has been done? It's a technology which is called HiC. I'll explain that in a minute. And basically from this technology, you can reconstruct how chromosomes fold. And that is basically a schematic. Each color represents a chromosome. Now, the amazing thing is that you have to reconstruct this, and the whole thing has to be, you have to be able to unfold it without making knots, obviously. Yeah? And this is what mathematicians then call a fracture globule. Don't really ask me what this is, but it just basically means you can fold something and unfold it very rapidly, and it doesn't make knots. Published in a cool journal of Knot Theory and its Ramifications. So this is where genomics really gets its novelty from. Okay, so that's the simple that we all know. This is how it really looks in reality. How can that be measured? And this is, as what I mentioned before, technology which is called HiC. I don't want to go into detail, but basically you take the nucleus, you cut the DNA in pieces, and then religate it. And because of the physical location at this moment when you fixate the nucleus, then you can reconstruct with the sequencing technology who was actually next to whom. Well, and who is next to whom is then represented in heat maps. And we like to look at these heat maps in these triangles, explain that in a minute, and you can also look at in this way. So one is from one viewpoint, shows which this viewpoint, which other pieces of DNA it interacts in the 3D room of the nucleus, whereas the other one is genome-wide. So what you can do then, you can even reconstruct from these maps a 3D conformation, like I've shown you before, of either the entire genome or parts of the genome, like you can see here. This is what we believe it looks like. So you have this blob here of interacting chromatin, and this blob would represent a triangle like this, or this piece of green interaction here. So this means this chromatin is tightly interacting, but it's not interacting with its neighbors, which would be the next triangle on each side. Okay? So that's the principle. Now, why is that important? One thing, it just tells us how DNA is really folded in the nucleus. But like in most things in biology, it's not just the folding, it has a major biological function. And this major function is that it's directly involved in gene regulation. So how does that happen? Because our genome is so large, the enhancers that regulate gene expression and the promoter, which is kind of the receptor of the gene regulation, they're very far away. So they have to contact in the physical space of the nucleus by physically interacting with each other. So that means this huge strand of DNA folds in the 3D space, and by that it brings the enhancers close to the promoter, and that results in transcription in a very simplified way. But this interaction and that 3D contact appears to be essential for the entire process. So that's, I think, is a major discovery and a major advance, and it tells us one other thing. So it tells us, these are then called, these triangles, topologically associated domains, and as I said, you can look at them from one viewpoint or genome-wide. So it tells us that these domains have a beginning and an end. That's the blob of interaction of chromatin. And they're separated by what we call boundaries. And across these boundaries, you have very little contact. So why is that important? Because it tells you when an enhancer sits here, it will regulate and interact with this gene, but not with the other one outside of the triangle, across the boundary. That's a very important information that we didn't add before, because in principle, these enhancers can act in any direction. So that means we have domains, and these domains are highly interacting, and they're separated from other domains that are then also interacting, but they don't interact with each other. And these types of interactions are interestingly super stable. So if you look at different cells, they more or less always look the same, because the domains are defined. And the other interesting thing is if you look at them in evolution, then you find, we do this all the time now, that you can actually put them directly one under the other. They're totally conserved, at least for the important developmental genes, for example. Yeah? It just means that these are blocks, it's almost like Lego blocks in the genome, that are then preserved in evolution. And for us, of course, it was the question then, so what happens if these blocks get disrupted in disease? And this is a nicer Hi-C with a high resolution, and you see these topologically associated domains. A big one then is a more unstructured region, the next one, and you can scroll through the genome, it just all looks the same, all different sizes of these interactions. So what would happen now if this boundary here, for example, or another one, would be gone by a deletion, if you would disrupt this? That's the question. And we had patient samples where that was the case. And then we studied this up and down with different constellations, and basically showed that this has a major impact on gene expression and is a mechanism of disease. And I just want to show you one example. So basically it's around this boundary here. So we ask if that boundary is gone here in the schematic, would this kind of spill over and activate the other gene? So what we have here, and that's again that region, we have here this FRNA4 gene and another one, a PAX3 gene, separated by these boundaries. Here's a strong enhancer. Now, in the deletion, this entire piece is missing, including this boundary. And here you see the interaction of the PAX3 gene in the wild type. It stays within its little tat. No interaction with the other one. But if we delete the boundary and this piece, then suddenly the PAX3 gene interacts with this enhancer that is actually supposed to regulate the ephrin gene. So what happens? Here it regulates the ephrin gene. Now it can interact with the PAX3 gene. That's the normal expression. And now we have an expression which is very similar to FRNA4, and that makes this kind of disease. Okay, so it adopts a different regulatory domain by the deletion of this region that includes this boundary. So another example is an inversion. So an inversion, you just basically turn things around, and the inversion is around this boundary. So that's the inversion here, the breakpoints. And here you have now the interaction from this Wnt6 gene. And now you see that the Wnt6 gene suddenly now interacts with that enhancer of the ephrin gene. So normally this would activate ephrin. Now it activates Wnt3. And what you get is again this type of expression which is not present in the wild type. And you get this very complex limb malformation. So this kind of principle has been now shown for all sorts of different diseases. It's something that is relatively rare, but it shows us how genomic pieces interact and how they are separated by 3D genomics. So one important thing and sort of technical advancement that really made all this possible is that we were able to create these large rearrangements in mice by CRISPR-Cas in a very fast system, so we can now produce these mice within a few weeks. It's really amazing. It's a little sort of factory that you can use. You basically cut and then it gets assembled. It makes either a deletion or an inversion or also a duplication. And then we can basically create anything we like, also in a very large sizes, right? Up to over a megabase. So that's was one thing. So what are we doing now? I think one super interesting question is that also most of the other, the focus of the past years was always is that we look at the genome and then we destroy things. We knock out genes, we cut things out, and then we say, because we destroyed this, we have now learned how it works. But that's only half of the truth, right? You don't really learn how it works until you are able to design something. And at the moment, no one is able to design a genome. So that's, at least in part, what we want to do. And what the cool thing now is that you can use, you can synthetically produce large pieces of DNA of 100 to 150 kb now in yeast, and you can basically, you assemble them in yeast, and you can basically design any sequence you like. So that, of course, gives us enormous possibilities. And then we have transferred this from yeast to bacteria, and basically then delete the piece in the mouse. Then there is a special technology to insert these large pieces. You insert them and basically can replace an entire large regulatory domain, and then look at that, how that affects mice. So we now want to identify sequences in the bat that make the bat wing, and then put these in the mice to what will happen in the future, and it's actually happening already now. And I think this is where genomics should go, is that we are really able to design things and not just simply destroy everything and then believe that we've learned how it works. Yeah, and that's it. That's the group a few years ago, and yeah, and I'm very grateful for all these different support that we've received, that you can see here on the slide. Thanks very much.
Thank you very much for this fascinating insight into modern human genetics. I'm sure there are questions. Andreas.
Have you studied to what extent these high-C interactions correlate with linkage disequilibrium?
Good question. I don't think so.
In terms of genome evolution, that might be an interesting issue to address.
I don't think so. At least I'm not aware of it. Probably has been done, but I don't know. Yeah.
Christiane Nusslein-Vollert.
In analyzing the TADs for unidentified human diseases, you focus mostly on bone structures. Why? One can do that with any gene, with any disease, can one? So why do you focus?
Why is that? That has.
And also in your engineering, you focus on.
Yeah. Well, yeah. There are several reasons for that. One is a very simple one. It's a historical one. So if you work with human diseases, then
can you go a bit closer to the
if you work with human diseases, they are all super rare, and you need international connections to actually get the patient. So that's a simple reason. The other one is that we always tried, and as you can see here, also try, is to sort of recapitulate this in the mouse. And then the LIMP is a fantastic model system. We know, basically everything of developmental genes. The phenotype is super easy. As you know, you have, stages of development where new things appear and where a lot of patterning goes on, and you can simply cut them off. And we can analyze them for gene expression, for chip, what. It's a wonderful system for that as a model system.
Dietmar Tanner?
Is it true that the non-protein coding DNA is the higher the complexer the organisms are? If you look at the whole evolution?
Yeah, we just discussed that. I don't. Well, let's put it this way. If you look at, we just discussed that at Ciona or Drosophila and so on, they have relatively very small genomes. But then humans, for example, don't have the largest genome, right? There are animals that have much larger genomes. Axolotl, for example, lungfish, and many plants have.
I didn't get it. Is it true or not?
No, it's not. Yeah,
it's not true.
Well, in some way it is, but it's difficult to generalize that.
It was published that way.
Yeah. In some way, it's true. Yeah.
Okay. Herbert Jeckle.
I mean, it's fascinating, these stats. But the next step would be to go on a single cell level. Do you think they can change? Because that would be a tremendous additional complication.
No, that has been done on a single cell level.
Yeah.
Then of course
but it could be done, right?
Yeah, it has been done. Yeah. And then the sequencing, information gets very sparse, but you can reconstruct it, and you can see the TADs, no problem. you can see that these boundaries are not quite as distinct as they are here, because there's more fluctuation in the whole thing. But in principle, you get exactly the same results.
Yeah, but can I ask a question to your question? Single cell analysis is great, but if you work on organ formation, you need the context of the neighboring cells, right?
Yeah, but we have to see what we look at here. So, and I think this is the very fundamental thing, and that is on first sight not so easy to understand, is that these TATs, in my view, they correspond to regulatory domains. So that means this domain is, whatever is in there regulates the gene that is inside. And if you then look at different cells, then the interaction within the TAT is very different, but the boundaries stay the same. And if you look at the gene expression in a fibroblast or in a hepatocyte, it doesn't matter, because the domain is going to be the same. And
but
that's evolutionary conserved. Yeah. The domain is the same. How the domain is utilized is different.
Yeah.
Right? That's the key.
I mean, my question is to be more specific. If you take stem cells, which can differentiate to A or to B, so if you look along the way of the differentiation, is it exactly the same?
It's pretty much the same.
Is?
Nothing is exactly in biology, but
no,
it's it's pretty much the same. Yeah.
Okay.
Yeah. 80% or so.
Regine Carmen has a question.
So if you analyze a family, father, mother, progeny, so recombination occurs, does not destroy the TATs. Has that been looked at?
Well, it doesn't destroy it, because it basically, you exchange the alleles, and the overall structure completely stays the same.
Okay.
So unless you have a mutation, as I showed here, but in principle, the alleles will only differ by SNPs, and this doesn't touch the whole thing at all.
Okay.
Yeah.
Okay, thank you very much. I think there is no further questions, so we thank you again and all the people asking questions for the very nice discussion.
Thank you.
And we come to the presentation of Jane Goodall's video. Jane Goodall is probably very well known to all of us here in the audience. She was awarded the Mendel Medal in 1987 for the groundbreaking studies of wild chimpanzees in Gombe Stream National Park in Tanzania, which forever changed our understanding of relationship to the rest of the animal kingdom. The transformation research continues today as the longest running wild chimpanzee study in the world. Jane's work builds on scientific innovations, growing a lifetime of advocacy, including trailblazing efforts through her international organization, the Jane Goodall Institute, which advances community-led conservation, animal welfare, science, and youth empowerment through JGI's Roots and Shoots program. She's a true icon, and I'm looking forward to the video she has provided to us. I haven't seen it, and let's just enjoy what she has to tell us.
Hello, everyone. I first want to say how really thrilled I am to take part in this honoring of Gregor Mendel on the 200th anniversary of his birthday. The Mendel Medal is one of my most treasured possessions. As a child, I was absolutely fascinated by Mendel's experiments with peas, and his life story also taught me that whatever the background, genius can shine through. As you all know, he was born to a poor family, and he became a monk so that his education could be supported. And he became a teacher, even though twice he failed the oral examination. And he became a gardener, and it was in the garden of the monastery that he performed his famous experiments with peas. And the other thing about this medal is that I received it 2 years before the unification of East and West Germany, before the Berlin Wall came down. And I remember so well being stopped by border police, crossing the Iron Curtain, passing fierce-looking police dogs, seeing the long line of cars going in the other direction, each one thoroughly searched in case somebody was hiding there trying to get across the border to freedom in the West. And I felt so honored that I'd been chosen by the scientists, who'd been isolated and cut off for so long from the Western world. And I was so pleased to meet them. I've just before sending this message been reflecting on how far the study and knowledge of genetics has come since Mendel's time. Back then, genes hadn't even been discovered, really, and there's such a difference between his experiments and the genetic modification and gene editing that are happening today. I really wish he would suddenly appear through the mists of time to tell us on this, his 200th birthday, exactly how he feels about these developments. Perhaps he would feel uneasy about altering genes. I have to admit that I do. So much has happened since I received that medal in 1987. After World War II and the horror of the Holocaust, there was a general feeling of relief and that this can never happen again. How tragic that we were so wrong. There have been countless genocides and persecutions and armed conflicts since then. And in addition, today, we're faced with climate change and loss of biodiversity, both of which we've brought on ourselves by our absolute disrespect of the natural world, by the crazy idea that there can be unlimited economic development on a planet with finite natural resources. And I only hope that gene editing doesn't lead to another problem, and as yet an unknown problem. We do know that we're taking our planet's finite natural resources in some places faster than nature can replenish them. And we know that we're destroying and polluting air, land, and water. Factory farms designed to produce cheap meat, resulting in shocking cruelty, and genetic modification that has, for example, caused some poultry, they can't even walk properly. They've got such fat thighs and breasts. So many people ask me if I really have hope after seeing so much destruction in so many countries, so much cruelty. Before I answer, I want to stress that for me, hope is not just wishful thinking, it's about action. I see our species as being at the mouth of a very long, very dark tunnel. Right at the end, a little star is shining. That's hope. But it's no good sitting at the mouth of the tunnel and wishing the star would come our way. No, we have to roll up our sleeves and crawl under, climb over, work our way around all the many, many problems that lie between us and the star. The good news is that there are people working on every single one of these problems, but so often, sadly, they work in silos. And sometimes one problem solved in one area can give rise to another elsewhere, because all these problems are interconnected. So we need to understand the big picture and get together and work in cooperation to solve the problems of the world today. One of my reasons for hope is the resilience of nature. Places that we've utterly destroyed can, given time and perhaps help, be restored. Nature will take over. Plants and animals will return. It may not be exactly as before, but still, nature will be back. I've visited many such places, and animals and plants on the very brink of extinction can be given another chance. Another reason for hope is the energy and commitment of young people once they understand the problems and are empowered to act. I began a program for youth, Roots and Shoots, in 1991, when I realized, as by then I was traveling around the world, talking about the problems that we were facing. And even back then, many young people, high school, university students, were losing hope. And if we lose hope, we become apathetic and do nothing. If our youth loses hope, we're doomed. So I asked these young people why they were feeling useless, helpless, apathetic, not seeming to care, or why they were depressed, or why they were angry. And they said, well, you've compromised our future, and there's nothing we can do about it. Indeed, we have been stealing their future, probably since the Industrial Revolution, maybe even since the Agricultural Revolution. The main message of Roots and Shoots is that every individual makes a difference every day, and we have a choice as to what sort of difference we make. There are now groups in 65 countries with members from kindergarten through university working on projects that they choose themselves to make the world a better place. A project to help people, one to help animals, one to help the environment. And it's through taking action that they manage to overcome their apathy, their depression, or their anger. Some young people, even now, have reached positions of decision-making, those that joined Roots and Shoots early, in the early 90s, and they take the values that they acquired in the program into adult life, the main one being respect for each other, respect for animals, respect for the environment. And my final reason for hope is the amazing human intellect. I spent years studying wild chimpanzees, our closest living relatives, both genetically and behaviorally. They're so like us. The major difference is the explosive development of our intellect. We now know that animals, as well as being sentient, are way more intelligent than science once believed. But they can't devise ways of traveling into space. They can't write books. They can't study genetics. Isn't it strange that the most intellectual creature to walk the planet is destroying its only home, planet Earth? I'm sure you've all seen those pictures from space of this tiny, precious blue and green globe surrounded by the dark, cold immensity of space. It's so precious. We must try and do our best to heal some of the harm that we've inflicted, to slow down climate change and loss of biodiversity. So we're now celebrating the 200th birthday of Gregor Mendel, a shining example of the human intellect. It's astonishing that it was 40 years before the value of his work was realized, although apparently he recognized that it would one day be recognized. Anyway, now of course he's regarded as the father of modern genetics. So Gregor Mendel, over the 200 years, I salute you. Thank you.
What a wonderful speech. I don't know if she's listening. It would be so nice to kind of transmit our gratefulness for this wonderful speech. Actually, this is the concluding remarks that is in the program. I think she said it all. And one of the sayings of Gregor Mendel was, "My time will come," because he noticed that he was not understood, and his time came, unfortunately, after he died. So let me thank Jane Goodall again and all of you for coming, but please stay put. We are not done yet because there will be some concluding remarks by our president.
Yeah, it's a very touching moment after those touching words from Jane Goodall. There's clearly nothing to add to those words, but one important aspect. I mean, you would wonder, what can a president who happens to be an oceanographer and paleoclimatologist say intelligently about a Mendel symposium? A few things, but not too much. So you may think it's trying to overcompensate with an oversized golden chain. Not really either. I wear this 6 times a year under usual circumstances. This year it will be 7, for a particular reason. And the one thing, when we prepared for this symposium, I looked at the list of laureates. And there's one person obviously missing amongst the Mendel laureates. And the only explanation I found for this person who's missing on this list of laureates is that sometimes there's a feeling, and I think Jane Goodall made wonderfully up for this, that if this person who should be on the list was awarded a potentially higher regarded prize. One such prize which we all respect as the highest prize possible is the Nobel Prize. So the person, the giant in the field, a pioneer, a hero in science, is Christiane Nüsslein-Volhard. So what we do now, and she doesn't know anything, and I think everyone kept quiet, liebe Frau Nüsslein-Volhard, and the Leopoldina does these days strange things which she's never done before. We do award you on this day of the 200th birthday of Gregor Mendel, a Gregor Mendel Sondermedaille, and I'll ask you to come to stage.
Die Urkunde sagt, die Deutsche Akademie Naturforscher Leopoldina, Nationale Akademie der Wissenschaften, verleiht Frau Professor Dr. Christiane Nüsslein-Volhard aus Tübingen aus Anlass des Leopoldina-Symposiums anlässlich des 200. Geburtstags Gregor Mendels für ihre herausragenden Pionierleistungen auf dem Gebiet der Entwicklungsgenetik, die Mendel-Sondermedaille. Alle an der Saale, den 20. Juli 2022. Das machen wir jetzt so. Die Ulla Bonas hat die Medaille. Und jetzt gibt es hier Herrn Scholz, der knipst.
Was denn?
Bisschen kippen?
Ja. Nicht, dass sie rausfällt.
Das ist keine Medaille, das ist wirklich für Sie. So, und jetzt. Und die Laudatio, das war jetzt eine ganz andere Choreografie, wird jetzt von Ulla Bonas als Nachklang gehalten. Und die Blumen, Frau Nüsslein, bekommen Sie auch schon. Jetzt genießen Sie die Laudatio.
What a surprise. Dear Christiane Nüsslein-Volhard, ladies and gentlemen, dear colleagues and friends. I'm extremely pleased that we end today's event with the award of the Mendel Medal to Professor Christiane Nüsslein-Vollard. And it's a great honor for me to introduce the award winner to you, although I'm sure that you know her or of her work, because Christiane Nüsslein-Vollard is one of the world's most distinguished developmental geneticists. Let me give you a brief overview. Her research on the genetic control of early embryogenesis in the fruit fly Drosophila melanogaster was groundbreaking for our understanding of developmental processes from gametes into an embryo with a complex body plan. For this, she was awarded the Nobel Prize for Physiology or Medicine in 1995. Since the early 90s, she focused her work on a new model system, namely zebrafish, to study developmental processes in vertebrates, more precisely the genetic basis of color pattern formation. Ladies and gentlemen, now I quote, "I want to understand how life works," unquote. That is one of her statements. Enormous curiosity has always driven you to get to the basis of things. Early on in her research, she combined developmental biology with genetics. Together with Erik Wieschaus, she generated a large number of Drosophila mutants and systematically screened them for defects in early embryo development. By microscopy, I mean, it's unbelievable, and I must say, not only by microscopy, but there was a lot of microscopy involved, and I always admired your patience and that of your co-workers, I must say. So this way, you identified crucial genes involved in the body plan of the Drosophila embryo, especially the maternal genes, which are transcribed into mRNA in the fly egg cell before fertilization, but these mRNAs are only translated into protein after fertilization. In recognition of Christiane Nüsslein-Vollard and Wieschaus' achievements, the Nobel Prize Committee awarded them, together with Edward Lewis, the Nobel Prize in Medicine or Physiology in 1995, as I already mentioned. Dear Christiane Nüsslein-Vollard, when asked in an interview why you started working with zebrafish, you said, quote, "We found a large number of very fascinating mutants from the genetic screens in flies, and I feared we would have to clone them all. So instead of having to do lots of sophisticated biochemistry, I thought that maybe we should do genetics, as this was much more interesting for me. I decided we should look at vertebrate development," unquote. So in the early 1990s, Christiane Nüsslein-Vollard turned to zebrafish and focused her research on the evolution and genetics of Danio ririo. That is the Latin name of zebrafish. In contrast to Drosophila, genetic mapping still had to be established in this organism, but thanks to her, zebrafish became a new model system for the genetic analysis of color pattern formation in vertebrates. One of Christiane Nüsslein-Vollard's central goals is to find genes that play a role in the variation of color patterns of related species in evolution, and thus to understand how diversity of patterns have evolved. Ladies and gentlemen, Christiane Nüsslein-Vollard knew early on, that is when she was 12, that she wanted to become a biologist, a passion she developed growing up in Frankfurt and exploring nature and spending vacations on the farm with her grandparents. After the Abitur, she briefly considered pursuing medicine, but dropped the idea after a 4 weeks nursing course in a hospital. Instead, she followed her genuine interest and started to study biology at the university in Frankfurt. In 1964, she went to the University of Tübingen and switched to study biochemistry. After her diploma, she got her PhD in 1974 for research on protein-DNA interactions and the identification of an RNA polymerase binding site in the replicative form of bacteriophage FD DNA. That was really in fashion also at the time, as we already heard from Regine. In the 70s, many people did genetics in E. coli and bacteriophages and found a lot of interesting stuff. But you switched to a higher organism. In 1975, Christiane Nüsslein-Vollert joined Walter Gehring's lab in Basel as a postdoc. Gehring was a specialist in developmental biology of Drosophila, of the fly. After that, she went to the lab of Klaus Sander at the University of Freiburg. He was an expert in embryonic patterning. And in 1978, she set up her own lab in the newly founded EMBL lab in Heidelberg, together with Erik Wieschaus. Over the next 3 years, they examined about 20,000 mutated fly families and collected about 600 mutants with an altered body plan. In 1981, she moved to the Friedrich Miescher lab of the Max Planck Society in Tübingen. And then 3 years later, so that means from 84 until her retirement in 2014, she was director at the Max Planck Institute for Developmental Biology in Tübingen. Since 1991, she has held an honorary professorship at the University of Tübingen, and since 2014 heads an emeritus research group. As visiting professor, she has taught at renowned universities in the USA, for example, Yale, Harvard, and Rockefeller University in New York. Besides the Nobel Prize, our today's laureate has received numerous other honors, of which I only mention a few, such as the Leibniz Prize of the DFG, the Albert Lasker Award, and the Federal Cross of Merit. If I counted correctly, she obtained 14 honorary doctorates, including Dr. Honoris Causa from Princeton, Harvard, University of Oxford, and the Ludwig Maximilian University in Munich. And besides her passion for science, Christiane Nüsslein-Vollert said that she loves to sing, play the flute, and do chamber music. And she published a cookbook in 2006. Dear Christiane, it is great to have you here today. With your innovative work on development and genetics of zebrafish, and in particular on pattern formation, you have broken new genetic ground for a second time after sensational results on Drosophila, for which the Leopoldina honored you now with a special Mendel Medal. Congratulations again, also from my side. Thank you for your attention.
So, the last word still has the president. And first of all, before I ask you to come back on stage, there are two more flowers. One, Ulla, you can stay. Is for Ulla Bonas. And the second one is for Regine Carmen. As our two main organizers of this wonderful workshop. Then I propose Frau Nüsslein-Vollert, probably with flowers, if you may, to join us.
Ich würde dich nur gerne herzlich bedanken und wollte noch eins sagen.
Moment, Frau Nüsslein-Vollert.
Ich möchte sagen, dass die zweite große Ehrung, die ich bekommen habe, war die Carus-Medaille der Leopoldina 1989. Das wird nie erwähnt. Das war für mich wahnsinnig aufregend. Das war kurz vor der Wende. Da war ich dann noch mal in Halle. Und dann würde ich auch noch gerne erwähnen, dass mein Urgroßvater Vizepräsident der Leopoldina war und Chemieprofessor hier in Halle, und mein Großvater war Medizinprofessor hier in Halle, und mein Vater ist hier aufgewachsen. Insofern habe ich eigentlich einige Beziehungen zur Leopoldina, und deswegen freut es mich umso mehr, dass ich jetzt noch eine zweite Medaille bekomme. Mein Großvater übrigens hatte die Cottenius-Medaille. Gut, ne?
Wir bleiben motiviert. Frau Nüsslein-Vollert, darf ich Sie trotzdem noch mal für ein Bild auf die Bühne bitten, vielleicht mit Blumen. Und dazu würde ich gerne alle Mendel-Preisträger für dieses Gruppenbild hier hoch bitten. Also, wer eine Mendel-Medaille gekriegt hat, möge kommen.
So, kommen Sie zu mir. Ja. So, gebe die Blumen aber nicht dahinter. So, und damit lade ich Sie alle zum Abendessen ein und vor allem auf ein gutes Glas Wein und ein Toast auf Frau Müsslein von. Vielen Dank.